Shall I start now?
Yes,
Uh, Good morning, everyone. Welcome to Swalife Biotech fourth annual International Conference 2026. My name is Ruchira Pawar, alongside with my co-anchor Ms. Nikita Varunski. We are delighted to be your host for today's conference. A very warm and pleasant welcome to each one of you. It is my pleasure to welcome you all to Swalife Biotech Annual International Conference 2026, a global platform that brings together researchers, academicians, clinicians, industry experts, and students to exchange innovative ideas in healthcare, pharmaceutical sciences, artificial intelligence, predictive medicine, clinical research, and drug discovery. Today marks the beginning of an exciting scientific journey where knowledge meets innovation and collaboration leads to impactful research. We sincerely thank every participant for joining us and contributing to this prestigious conference. I am Ruchira Pawar, and it's my privilege to be your host for the first half of today's conference. Without any further delay, let us begin our first scientific session. It is my great honor and privilege to introduce our first keynote speaker, Dr. Praveen Bade sir. Dr. Praveen Bade sir is an accomplished researcher, educator, entrepreneur, and visionary leader in pharmaceutical sciences and predictive medicine. As the founder of Swalife Biotech, he has been actively working towards integrating artificial intelligence with biomedical research and drug discovery, precision medicine, and healthcare innovation. Through his dedication to scientific excellence, he has inspired researchers and students to explore the limitless potential of AI in healthcare. Today's keynote lecture is titled Predictive Intelligence in Drug Discovery. This session will provide valuable insight into how artificial intelligence is revolutionizing modern drug discovery by accelerating target identification, optimizing lead molecules, predicting toxicity, and supporting precision therapy. Everyone, please join me in extending a warm round of applause to welcome Dr. Praveen Bade sir. Sir, may I kindly request you to take over the session?
thank you, Ruchira. Uh, can you hear me? Is my voice clear?
Yes, sir, we are audible.
Okay. Good morning, all, and welcome to our fourth annual um, Swalife Biotech Conference. Every year, around July, August, we organize this conference. The purpose is to actually have the interdisciplinary keynote speakers and simultaneously talking about what Swalife is doing and how we can actually contribute, innovate, and give the value which can be useful in the further drug discovery and development. So, as Ruchira mentioned, in today's keynote speaker, I'm going to talk about. Just I'm going to talk about predictive intelligence. So basically, the focus of Solar Biotech um, is towards preventive formulations, but simultaneously, the increase in the large language models, that is the AI, the predictive drug now, predictive medicine is now, leading towards the market, and we believe that there will be, change in the drug discovery and also in the healthcare due to the AI, due to the large language model in drug discovery and simultaneously in the development. We are moving further one step. That is not we are not only sticking to the predictive intelligence, but I will be also going to show you that where we actually lead after predictive intelligence and how that is helping not only in the drug discovery but also the researchers, the doctors, and also the pharma companies who can actually utilize the model for developing preventive, predictive, and precision medicine. So in this session, as Ruchira mentioned, I will be talking about drug discovery, but I decided that I will actually include early diagnosis as well in my study. So we are not only in the step of predictive analytical, but we have moved into predictive decision medicine intelligence, which actually helps the researchers, the doctors to take the decision based on the evidences, and that is what I will be talking about. So majorly, when we look at the prediction, having evidences and transferring that evidences in the decision is where actually the further drug discovery and the healthcare is going to move. So when we look at the key challenges that we have in the drug discovery and in the healthcare, is not about the discovery, but it is about predicting early. That is actually having early diagnosing, having early information about the disease, so that way we can actually treat, or we can actually have uh, the dosage form ready for that phase of the healthcare. So healthcare actually has a prediction problem, where the major question is that can we actually have a formulation that is predictive one and not a reactive one. So having a diagnosing of the disease which will be occurring before leading to a disease stage or towards the advanced stage, that is where actually the predictive medicine work. And when we look at the model of our drug discovery, which normally takes 12 to 15 years, it is more about treating the disease which has already been occurred. So there is a chances of actually only maintaining the symptoms of that patient. um, Recovering the patient from that disease is actually negligible, and there are no actually such type of work where we are actually looking in the treatment. So when we look at the drug discovery, having a predictive model in the drug discovery is going to help to reduce the disease burden, and it will be a useful model for further healthcare. So the central question which I want to answer today is that can we reliably predict early rather than actually reacting at the late? So instead of patient coming in the surgery and then we are having a symptoms which are going to be diagnosed and then the treatment is given, can we do it early? So as a researcher, that is where we are. As a doctor, we want the doctor to actually take a decision when they are treating a certain diseases. So where does the predictive come there? When the diseases has occurred and when the treatments are going on, many times there are other symptoms, there are the toxicity developing due to the treatment, which need to be then um, look into, and that can be done with the predictive model. So, what is predictive intelligence? So, when we look at the predictive intelligence, it is about the data which is already there in the public domain, simultaneously the symptoms which are being, done given by the patients. Simultaneously, the predictive models and the generative models databases can be utilized, which will be collected as evidences. Now, with that data, with the help of the AI models, you can actually put them into a framework which will be a structural databases, and that we can call that as a graphical structural databases, which will be helping to develop the context, that is about the biology of the disease, about the population, about the scientific state of the knowledge. And with the help of that, you can predict that what actionable intelligence can be utilized from the data which has been put into a structural format by the AI. So that way we can actually design In this case, the researcher can design the experiment. The further experiment, it can be in vivo, it can be clinical, or it can be actually observational clinical trials. And then based on that, we can move faster towards the development of the formulation. So that is where the predictive intelligence is going to be playing important role and will be useful in the further drug discovery and also in the decision making. So now with that, you can actually move towards data to decision. The data is going to give you the signals, it is going to give you the records, which, as I told you, will be put into the structural format, and that structural format help in the knowledge development. Now all this is already happening, but major of the thing is in the fragment form. That is, you have to go to certain databases, then from there you have to develop the graph, and then from that graph manually you have to develop the relation. But all that can be now with the help of the AI. You can do it with large language models, but it doesn't mean that when you do that with the large language model, it is going to be the final answer, because there will be still the knowledge from the scientists who actually have that critical thinking and scientific thinking, which can be utilized for making decisions. So you take the assistant of the AI large language model for the prediction purpose, but the decision will be made from the data, from the structural data, by the researcher, by the scientists, by the pharma companies, by the doctors. So that can be taken into further in the in the treatment, in the research, which will be creating as a learning. So that moves us to the predictive medicine, which is actually changing the healthcare model. As I mentioned early, instead of reactive, you will be proactive, where it will be majorly predictive and personalized medicine. So we are already known about the reactive model, where the disease has occurred and then the treatments are going on. We also know the model of prevention, where we are actually screening the population, and based on the biomarkers, based on the targets, we are actually helping them to prevent the further disease occurrence, or even the further issues with the same disease which has been in the stage 1 or at the primary stage. But with the predictive model, what is going to happen? You are going to uh, treat the individual before actually disease is going to occur, with the help of the data driven, that is the evidence driven insights, which are already being utilized from the public domain, from the other patient symptoms, and also using the predictive model of the large language model. In addition to that, you can also utilize those for the personalized or precision formulations, which will be helping to understand. And that where the doctor's predictive intelligence is useful, because they will be then, instead of judging, or instead of taking decision based on what is available, they can actually do the right intervention, which will help the right patient to target the disease based on the biomarkers, based on the targets which they have been having issues in that disease. So that is where the personalized, the precision medicine model can be moving into. So the question now here comes is that when we're talking about predictive intelligence, where actually this predictive intelligence comes in the patient journey? Because when we are looking, when I started with the predictive intelligence in the drug discovery, where the drug is still at that stage where it has to move into preclinical, clinical, and also into through the regulatory in the market. But where actually it comes into the patient journey, so it is not the single point. It can actually be in different areas, and that's why I mentioned about early diagnosing as well. The predictive intelligence can help into the population level signals. It can also identify the risk. In the early diagnosing, you can actually identify multiple biomarkers or the different proteins with the help of the imaging system. So that is another area. Then you can also understand the targets in different subgroups. So when we look at the patient having the same disease, but they can actually have different targets which are been actually causing that disease. So molecular and clinical data is going to help with the help of the predictive intelligence. And based on all this, the doctors can actually have a evidence guided formulation created for that patient. So that is again the personalized or precision medicine, which will be then continuously reassessed for the long term cognition modeling, that is the monitoring of the outcome, which will be helping the patient. And that all comes into the predictive intelligence. So you need to understand that when the researcher is working on a project, they are critically evaluating the literature, and based on they develop the hypothesis. When doctors are also treating the patient, they are again doing the same thing based on the symptoms, based on their experience conclusion, they decide on the treatment. But with the predictive intelligence, you are going to have a evidence that yes, this hypothesis need to be tested, and we are not only taking the decision based on our critical thinking, but simultaneously we have a research assistant, that is the large language models, that is AI, which is also helping us to make sure that we are moving into the right direction with the right prompt engineering information, which will be useful to take the research further. So that is where predictive intelligence is useful. And in the patient journey, we have seen all these four areas. Now, when we look at the early diagnosing, there is already a lot of data available from genomic, transcriptomic, then metabolomic, which actually gives us the information about the disease signals. In addition to that, we also have different images of the biomarkers, which actually give us the features which will be helping us to validate the early detection. uh, with all this data which is already available in public domain, if you connect that with the medical record, that is the electronic health record of the patient, that can be medication, that can be comorbidity, and in addition to that, any clinical evidences which have been published, that will help us to validate the model, the AI model, which will be utilized for the predictive intelligence in the early diagnosing. So that is where actually predictive intelligence is going to be very useful. So when we look, we recently actually working with one of the UK university, working on one of the project, that is early cancer diagnosing. So now when we say early cancer diagnosing, we are not talking about when the actually the cancer has already occurred. We are talking about the biomarkers, we are talking about the data, clinical data, where the person or the signals has been already been shown into the cells, into the biomarkers, which can lead towards the further cancer development. And there where the early diagnosing come, not when actually the patient has already have the the cancer cells and they are growing, which can be treated with chemotherapy, but still they will be coming back again. So that is not the early diagnosis on before actually having the onset of the disease, and that's why predictive intelligence is important in that phases. So now with the biomarkers, as I mentioned, you can actually utilize the imaging and also the clinical observations, which are important, which are sensitive, specific, and also reproducible, utilizing different assays. So with the help of that, you can actually understand the disease risk with the help of the predictive intelligence. So while talking about all this, I am actually giving you the theory of predictive intelligence, where we have already developed a predictive intelligence model which actually work on getting information from public domain utilizing the large language models, which I will be talking later. Now, with the help of that, you can develop the statistical and machine learning model. So we are into that stage where machine learning models has been developed, and we are utilizing also the information from the public domain and with the groups of our collaborative doctors, the high risk patient identification and early intervention. So that way we can actually make sure that the biomarkers which has been identified are actually leading towards the disease. So that way the predictive can be identified at the stage where disease can be prevented, disease can be treated early in the stage where there is no onset of the disease symptoms. So, as I mentioned, that we are utilizing the large language models and the data which is already in the public domain. With the help of our network of the doctors, we are actually having the clinical data. So, this is the layout that is working with the predictive intelligence, where the genomic, transcriptomic, proteomic, and metabolomic data from the public domain and the clinical data which is at the moment there in the patient are being utilized, and with the help of the large language models we are actually having a predictive intelligent model which can be utilized in the decision making. Now we will go back to the stage where predictive intelligence start for the researcher at the drug discovery stage, where the target identification, hit and lead identification and validation, lead optimization, and preclinical work can actually be done with the help of the predictive intelligence. So now, while talking about clinical research, while talking about the patient, why we came back with the target identification is because in the drug discovery, the major part has to be done at the initial stage. The drugs, the molecules which are losing in clinical research is not due to the clinical trials. They are losing or they are coming out from the clinical trial because of the initial drug discovery process, where the targets, where the leads are not properly identified, validated, and not properly studied in the preclinical stage. And that is where actually the predictive intelligence is going to increase the speed, but simultaneously it is going to make sure that it will transit those target, those lead into the further drug discovery process and development process by utilizing the evidence driven prediction that can be give us information about de-risk go or no go decision. So that way we can prioritize only the promising candidate, and also making sure that the right targets has been taken into the drug discovery, so that way it can reduce the late stage failure. So, as I mentioned, predicting the right targets are important, and literature mining, literatures, literature survey and literature mining is a major stage when we are looking into the drug discovery, and that is going to help us to extract the important network, important association between the different targeted targets in the disease, different genes in the disease. So that way we can actually make sure that they are put into a proper structural database. I mentioned earlier that is the knowledge graph, which actually shows information about the target pathway and the disease. So that way we can actually have that confidence level that what is already been studied and how it is connecting to the different pathways. And then we will have the genetic evidences which actually give us the information, so which type of pathway analysis need to be done, so we can actually have the right targets in the further drug discovery process. Then comes the prediction of the molecule. Now the molecules which are already there. In case of Swalife Biotech, we are focusing on small molecules of plant because we believe that the active molecules from plant are going to targets different multiple targets. Majorly when we look at the allopathic drug at the moment, a single molecule is targeting single protein, and due to that the treatment is difficult because the body actually is having different proteins and they are mutated, or there is a problem with different protein when the disease occur. And that's why we have to have multiple molecule targeting multiple protein when the treatment is going on. So with the help of the predictive molecules, you can actually have the information about the molecule before you move into the in vitro or in vivo in the lab to test them. So QSR and molecular properties can be studied with the large language models, and also the tools which are already available by designing different analogs or designing different novel molecules. ADMET production can be done, then molecular docking can be done. Now, this was already possible, but now with the help of large language models, the predictive AI and the generative AI, you can actually have de novo molecule generation, which can be tested early and in silico for the desirable pharmacology and safety profile. So that is where the process is moving now with the drug discovery stage, utilizing the large language model in the predictive intelligence stage. Now in preclinical development, like designing the in vitro in vivo studies to study the efficacy, you can utilize the predictive intelligence, understanding the toxicity and safety. In case of Swalife, we actually believe that the molecules, the formulations, moving first into testing the activity, we should simultaneously have the toxicity and safety profile of those more formulations and the molecules. So when we are working on the our chemopreventive molecules and formulation, we also have skincare and hair care formulations, but we had made sure that the toxicity and safety studies are done on those all formulation to make sure that it is not only bioactive but it is also safe for the human use. Then PK/PD modeling is also important, and that is also available utilizing the predictive intelligence. That is the one of the role of the large language model, which helps to make sure that you can transit to the relevant model of in vitro, in vivo, and also if there is no need of in vitro, in vivo, you can directly move into the clinical study. So I'm talking that about for the herbal or nutraceutical type of the formulation. So one thing which I want to highlight here from all the presentation at the moment which has happened till time is that when we're talking about predictive intelligence, it is working as a research assistant. We are not replacing any scientist or any critical scientific thinking of the person. So human is very important here. Running through a model of AI is going to give you content, and that is where at the moment everybody is there. But moving into the decision making, moving into understanding the data is where actually the product of Swalife is going to work, and that is where I'm taking you later. So why AI is not enough? That is what I mentioned in early slide, that it is a model where you can actually have information which is in the fragmented type. So to give you an background of this, if you know the large language model, different large language models like ChatGPT, Gemini, Perplexity, Queen, DeepSeek, Cloud, all these models, when they are run for a literature search, they are going to give you different information, and we have already tested this. We have got one single product allocated to 10 of our research internship student, and we have collected the information, analyzed that information for the same project, and all large language models have given us different information because they are predictive and generative models. So you have to remember this. So it is very important that when you are doing the literature search or you are working on a literature, you should always use a RAG model, and that is going to help you with the information by giving the citation of the articles which are there in the public domain. So with the large language models, they are the fragmented tools. So literature search, prediction, or analyticals exist separately, and they are not communicating to each other. Simultaneously, there is a hallucination of the information. So if you know that ChatGPT can generate the reference preferences for the information which is not there in the public domain. So that is again something which need to be taken care when you are utilizing the information from large language model. So the output can actually have lack of traceability, lack of provenance, because they have been generated by the AI. So there will be difficult when they have been utilized, because sometimes it is a predictive information which is put into a structural scientific context, but it is actually reducing the actionability because it is not traceable, and there is a problem with the scientific accountability. And there where the senior researchers, the experienced guide role comes in, and it plays important role that actually you should review, you should monitor the information given by the large language model. So the predictive intelligence which I'm talking is actually a knowledge layer of our decision making, which makes sure that we collect that and then we lead towards the decision making. And I'm going to show you that transition as well. So just now I said the same thing, that we collect the information that can be a predictive one from the large language model, and then we take it towards the decision intelligence. But we make sure the fourth step, that is the validation, the evidence-based verification, is being done by the scientific researchers, so that may the content which we are validating is from the research, is from the public domain, which has been published. So predictive intelligence is not simply using AI to predict an outcome. Remember this, it real value is when actually the prediction is connecting with the scientific evidences, and then that scientific evidences are transfer in the clinical decision or in the research. So you need to remember this, that we are not utilizing the large language models or AI just for the predictive outcome, but the predictive outcomes are utilized for connecting with the scientific evidences, and then we are transferring them into the decision. So decision stage is important now with the product of our Swalife. So this is the now the second stage, where the platform that we have created is utilizing the prompt engineering to talk with the AI, that is the large language model. So that way we can collect the scientific evidences, or we can collect the predictive or generative information from the large language models, and put them into a structural graph which will be reviewed with the scientific data which is available in public domain. So that is where the predictive intelligence platform comes into the picture. So the core structure is to actually make sure that the large language model, the AI intelligence, is used in the early drug discovery phase, which will be helping to convert the information from public domain to the predictive medicine. So when I'm saying predictive medicine, that means the doctors or the researcher can take the decision to take it into the further studies. It can be preclinical, clinical, or in the market, by developing the formulation. So if you are working on herbal nutraceutical, this is very useful, because that way you can develop the formulation and it can be taken into the in the market. So now I will introduce the layout of our Swalife predictive intelligence platform, which actually helps you in the literature mining. It helps you to connect that literature of that project into a AI discovery, from basic target identification till IPR. Then the literature in mining and the AI discovery content is then being analyzed using the predictive analyticals, which help us to understand that what is available about the project which we are doing. And not only that, it also gives us information, the predictive information, generative information from large language model. So when I'm talking about this predictive or generative, as a researcher, you need to remember uh, that when you do a Google search, let's say we've done a Google search for DNA damage, or just any term like cancer, what is happening, the Google was giving us the different website on which the information is available. Now, similar thing about the large language model, about the AI, is that it does the same thing. It is actually giving us the information which is generated or predicted. It sometimes also connect us with the information which is there in public domain if your prompt is asking that connect me to the literature. Now the RAG models like Perplexity, they can actually directly connect, but simultaneously they also make sure that they generate and predict, and that is, we have also tested that um, so so the predictive and generative content will be there from this large language model. So when we are doing the predictive analyticals, we will be having content which is there from scientific evidences, and we will also have content which is actually predictive and generative from the large language model. So based on that information, we move to the fourth stage, that is the predictive medicine, which is going to help to understand that if I want to develop this project as a researcher, to take it further into the clinical, to take into the formulation, or take into the market, or actually work on the animal studies, I will have that information, and that will be with the help of the prompt engineering and with the help of the large language model. So what we have built, so when we started 2022 with the large language model, we understand that we are moving into the stage where instead of doing a Google search, now it will be on the large language model to get the information. But again, as I told you, it will be majorly generative and it will be predictive. So all this large language model information is actually generative or predictive. Some of the RAG models are connecting you with the databases. But when we look at what we already got in the research, if we think about the scientific intelligence point of view, we have got different databases from where we need to do the literature search. We do the literature search, we collect the literature, and we read it for scientific critical thinking purpose. But now with the help of large language model, we can utilize them for reasoning. So instead of looking into the article more deeper, with the help of large language model, having that 5 to 10 questions which will be helping us to understand that article, we can actually give that to ChatGPT or Gemini, and we can give the prompts that will help us to understand. So that is the second stage we can do. With the help of that, we then go for in silico study, and with the help of in silico study, we can then move into, if you're if you're from the chemistry background, you can go for novel synthesis or analog synthesis, and then testing them on the you can add plant herb, you can also add small molecules, the one which are from plant. If you have synthesized, you can actually put the molecule which is similar to your similar category of your novel molecule. If it's analog, you can put the molecule which is which you have uh, create the analog of. Then the disease which you are working, if you are creating a formulation, you can put formulation, you don't have to put nothing is compulsory here, but it is good that if you put all the information, because this is what is going to utilize for your prompt engineering drafting. So that way it will have a prompt which will be utilized in the getting the information from the large language model. Also, we have put the regulatory reason, so that way you actually got different reasons which you are designing the project for. Target population, study type. Now the study type we are majorly having is the literature review and the evidence structure, and then you can put your project name. If there is any other information that you want to put, you can put here, because if you're already having some of the information which you are doing on that project, you can put that here. Once you save that, this project will be saved, and then you will have the four modules which will be running in the predictive intelligence, which is going to give you the information, and we are going to talk them in the further slides. So as I told, you are going to add the information about your project, that is plant molecules, disease, the formulation, and the population. So this is not working here. Okay, so I think the PPT I need to share. So I will just share the screen now because I want to actually show you each stages of our predictive intelligence. Okay, can you see my screen? Ruchira, can you see my screen?
Yes,
Nikita?
sir, we can see now.
Okay, okay. So yes, so when you start, you put all the information about your project, and then you click on the first module, that is the literature mining, which help you to extract the information about your project by designing the scientific prompt. So that way you collect the evidences, identify the gaps, and with the help of that evidences, you move into the further stage. So this is the the layout of your literature mining, where it actually have the complete evidence pack for understanding the molecule or the plant and the disease relation. Simultaneously, if you're working on plant, the phytochemistry, the target, and the safety and the regulatory for your molecules, the plant, or and the disease. You create the prompt using the tool. Here is the first stage, and you run that in any of these models, that is ChatGPT, Perplexity, Gemini, Cloud, Grok, DeepSeek, and Queen. But we have now come to a conclusion that Perplexity is where we will be majorly utilizing. Yes, Cloud, Gemini, and even ChatGPT. Now if you put in your prompt that search the live web, definitely it also gives you the live web. But initially when we created this in 2022-23, we were majorly focusing on the Perplexity. And once you got that information, you paste that information here and you save the run to convert this into a database by giving again the scientific prompt entry, which is generated utilizing the predictive intelligence. Then from there, you collect all the information publication regarding your project, and you put that evidences in the JSON file. The JSON file is a structural information which is actually machine readable. So when we are uploading our information, the data on our databases, we need in JSON file, because the normal Word file and also the HTML files, HTML file is also okay because they can be put into the structural format, but with the JSON it will be machine readable and create the knowledge base. Now, in the second stage, as I told you, it moved from target identification towards the study designing, where you actually have evidences about different target. Now, again, you have to remember that when we are running the scientific prompt utilizing the prompt design in the predictive intelligence in large language model, you will have evidences and you will have generative information. So even with the target, even with the pathway, you need to make sure that they are evidence based, and hypothesis generation is connected towards the evidence base. So that way, whatever the large language model have given us in the study design need to be made sure that it has been recommended with the help of the experiment which are there in the databases. And this is the second module where it moved from target towards the regulatory. Even the IPR and marketing information about your project is also collected from the databases, and also with the help of the predictive and generative you can also have that information connected to this predictive and generative, so that way you can have a novel hypothesis or the gap which can be identified. The third stage is the predictive analytical. So once we got the information, the literature from the data mining and from the AI drug discovery and designing, you understand the pattern of the information which is hidden and associating with your formulation molecules or your herbal. So that way you can connect it with the right studies which you want to take further. So that association need to make sure that it will predict the information which can be created for preclinical, clinical, or in vitro studies, and you can prioritize your research based on the information, the evidences which are strong or which can be translated into the relevant studies. So that is where actually the predictive intelligence comes. So again, with the predictive intelligence, you can see that the interfaces where the synthesized literature and the discovery scores are fed into the predictive analyticals, and the both uh, the literature mining and AI reports are utilized for the predictive analytical purpose. And that information is taken further into the understanding of the predictive medicine, which is a information about the project, about the data which is there in the public domain, which can be utilized as a development of a formulation or for the preclinical or clinical studies. And based on that, you can actually have the evidence-based content which can be utilized by the researcher, by the doctor, for the further studies. So that is where this predictive intelligence is useful, and you will have this type of the interface for predictive medicine where personalized stratification, monitoring, safety first guidelines are given based on the information from first three modules, and you will have information about your project in the different category if you are planning to do a research in different areas. So when we ask a research question, we actually have multiple models, but with the help of our predictive intelligence, our platform, we are going to structure the scientific problem with the help of the prompt engineering, which help us to curate the evidences and define the question precisely. So that is the important role of the model of our platform. We utilize then the large language models for reasoning purpose, for synthesis purpose, and for knowledge retrieval purpose, which is a powerful state of art model. Now, again, you need to remember that the purpose of the large language models are to make sure that we are going to reason, we are going to synthesize, and we are going to have the RAG model, that is the RAG powered models which are going to connect that with the database. So that way, when we have the output, it will be multi-model response from the evidences which have been compared, synthesized, and traceable. So that is where the predictive intelligence of Swalife is working, and it is not just a platform which is completely like utilized for LLM, but it is making sure it is giving you scientific useful information which is lacking when we are actually doing it manually. So this is one of the example where routine molecule was actually utilized in a project where we looked into diabetes 2 for oral capsule in the adult of 40 to 60. And what we have gone through is that then the literature mining, which help us to extract information about routine and diabetes. Then it connect the disease biology, the pathways to the targets, and those targets and pathways will understand the interaction between like protein-protein interaction, protein genes or DNA interactions to understand. And based on that, the population specific safety profile, because the molecule is from plant, you can directly move into the preclinical and directly into the clinical phases based on the information which you have got regarding safety and also bioactivity. So that structural research hypothesis is created with the help of the predictive intelligent workflow. So what the platform produce? It produce the evidence summary. It gives you the hypothesis and insights. It helps you to design the studies which will be moved into a structural report and a JSON format data, which is a machine readable evidence record. Now again, based on this, you can go for manual and you can start your wet lab. That decision you can take, but we decided that we should actually have a decision layer as well, and there where actually these reports are being utilized by converting them into JSON and feeding to the machine learning model. So the missing layer which we saw in this was that project one. So in project one, we done routine, look for the evidences, we have the report. In project two, we took project B, we have quercetin, we have the evidence. But project A and project B are not connected. Now we done a project C, and again that was not connected. So how do we accumulate, validate, connect, and reuse that intelligence across hundred or thousand of study? Was our critical question and was our critical limitation of our predictive intelligence model, because predictive intelligence model, the predictive intelligence was not having the memory. So that intelligence cannot accumulate and learn from itself. So it is not going to be an intelligent system because it's not learning, it is just giving you the information. And that was our missing layer, which we decided that there is a need to put a memory into this. So the AI output is not the same as organizing the knowledge. So the question when we had from the predictive intelligence is, after having predictive intelligence, is was that what if every research project, every paper we read, or even the hypothesis we generate, or even the experiment we conduct should contribute to continuous growing evidence intelligence system that can actually improve the next decision by learning from its data. And that is where actually we have the product called predictive and decision medicine intelligence. So it is not only going to predict, it is also going to help to take the decision. Now, if why to take a decision, we can do that scientifically by looking into the predictive intelligent report. Yes, but that will be only for one of your project. What about if you're working on numbers of project like what Swalife is doing? We are working on different molecules, different plant, different analogs, and all that data is fragmented, put in different folders. But with the help of predictive and decision medicine intelligence, now we got a complete database where there is a knowledge layer which actually connect to the decision layer. And I'm going to talk about that. That is the predictive, that is the PDMI knowledge, evidence intelligence for predictive medicine. So it is converting the structural scientific evidence into a knowledge graph and for a research decision. So it is not actually just a product, but it is a evidence memory which helps into the decision layer of the unified system. So this is the layout of the first minimum viable product of our PDMI, which is a evidence intelligence for predictive medicine. That is what we are calling. We have tested it with 10 projects, and we already have started putting the evidence layer, which also shows us the research decision. I will run the demo after I go through all the information. I will show you how the PDMI knowledge work. I will also show you the predictive intelligence layout as well. So, predictive decision medicine intelligence is a evidence memory and also a knowledge infrastructure, which is connecting the intelligence that is generated by the predictive intelligence platform, which is continuously growing by collaborating with the different researchers, different academicians. We are reviewing them the reports at our level, and then we are putting them into our decision platform, which is then being utilized by the PDMI for the researcher, for the doctors, and for the pharma companies purpose. So it is a predictive intelligence layer which actually generate structural evidences. So now in this decision making, it is not generative, it is not predictive like what we have in the large language model. So now you need to remember here is that what we actually got in the decision layer is all from the literature, from the databases, from the in case of the doctors, it will be symptom based as well. The symptoms which the patients are giving, they will be connecting to the information which is there in the databases. So that knowledge layer and decision layers are there in our PDMI. So why PDMI? And I mentioned earlier that there are already different databases available which are in the fragment form. So knowledges you need to get from, let's say, Google Scholar, you get the literature, you get from Scopus. So the papers, the report, the databases, all these are in the scattered format. The outcome of AI also is disconnected. So we connect that in the PDMI, and we rediscover the problem that researchers are having, that is the repeatedly regenerate evidence that already exist elsewhere. So many of the time there are research work we are doing which is already been done by someone and also been published, which has not been come into our context. So each project start from scratch, then the intelligence from the past decision does not actually help us in the for that scratch project. So that is again no accumulation of the information. So that's why we actually have this PDMI for the researcher, for the doctors, and for the pharma company purpose, which helps us to get the evidence sources, extract them, put them into the structural JSON format, which are then reviewed by the researcher scientist, and then it moves into our decision layer. So that is the architecture of our PDMI knowledge layer. And the first stage of that knowledge layer is the evidence extraction, which are done in the predictive intelligence, where compound targets and the information is extracted, the relations have been established. Based on that, we also identify which methodology or results can be taken further or can be searched, and based on the limitation we can decide on the studies which we want to take. So that is the evidence extraction layer, which are put into the knowledge building. So I showed you in the in the platform, you start with the knowledge project. You start with the knowledge project. So that is the knowledge project where you upload from the reports from the predictive intelligence, you review them, and you build the knowledge graph. I'm going to show you the knowledge graph as well. But the main feature which we have got there is the human review, which is very important, because based on the information that you have got from the public domain and from the AEI, you need to make sure that it is not generative or predictive. And that's why we also have a rule-based validation. We have both the models in the PDMI, that is rule-based and also the AI-based. We also connected that to the AI. So you can actually have that information, but that has to be reviewed by the human, and that review only goes to our our decision model, uh, the PDMI main model, which will be utilized by the researchers, by the doctors, and by um, the pharma companies. So the knowledge base has already grown now about 50 plus project, and we have got more than 1K of the source document with more than 10K of the entities and more than 25K of the evidences. The model which I'm going to show you is the basic one, but we have moved further into the the updated knowledge. So I think I will directly show you the both the platform, because it's already 10:30 now and we need to start with our next step. Okay, so I think I will just show you uh, the platform, because we're already like 1 minute late or for the next. So I'm just give me just 2 minutes. I will just use that 2 minute to show you so this is the predictive intelligence model. As I told you, literature mining, which will have two layers. One is to take the complete evidence and put them into the tables in the structural format. What happens is that when you load this layer, it is going to give you the structural prompt for the extraction purpose, and what we want, we have put that into here. So as per your project, it is going to change. So let's say for for complete evidence pack, it is going to act as a scientific analyst. And when you change the layer 2, it will be now going to act as a data curator, so collecting the information, and the data curator is going to convert it with the help of the scientific prompt. So that is where the predictive intelligence is there. Same thing with the second module. You got three layers here. One is to target, one is to design, and then it put into the structure. Then in predictive intelligence, you connect the both the layers from literature run and discovery run, and then the prompt is created, which will be then utilized for predictive analytical. And the fourth is the predictive, which medicine, which only have got one, which actually utilize the information from the analytical model, and then it converts the prompt into the predictive medicine purpose. So that is how the predictive intelligence work. And then we are into the main the platform, that is the uh, PDMI, predictive and decision medicine intelligence, where you got knowledge builder and the research builder. The knowledge builder is to actually put into the information to the question, the tool. And the research decision is for the research purpose. You can actually have combined knowledge graph, which is the network. You can focus on any of the work which you are doing. So let's say if I'm focusing on apoptosis, the knowledge graph is going to change to apoptosis. So there is a database available, curcumin, modulating the apoptosis pathway, and how it is doing. You are going to get the relationship here when you click on the bar which is connecting. So that is how it works. Then in the research decision, you can work on, let's say you decided that I want to work on oral cancer. You put here oral cancer and you ask to generate the the decision. And based on the databases, you are going to have current evidences. You will have evidence strength available, strongest support relationship, evidence maturity, then mechanistic coverage, and the major research gap. That is very important, because based on that you are going to suggest that what you will start in the cancer research. And again, there are other things as well: research priorities, question requiring experimental validation. So all this is helpful for a researcher at the initial stages, and also the researchers who are working on developing different formulation. And that is where we are utilizing it for our formulation development into skincare, hair care, chemo prevention, and also in the oral and breast cancer treatment purpose by developing different analog. Thank you for your attention. Sorry, I have taken extra 6 minutes. I now hand over to Ruchira. Ruchira, if there are any question, I think we will start with the presentations. If there are any question, I will answer them later.
Are there any questions? So thank you so much, sir, for such insightful and inspiring keynote address. Your session has beautifully highlighted how predictive intelligence is transforming future of pharmaceutical research and healthcare. We truly appreciate your valuable time, guidance, and expertise. We shall now begin our first session, that is poster presentation session. Before we invite our presenters, I would like to acknowledge and introduce a panel of evaluators who will be assessing today's poster presentation. Their expertise, experience, and valuable feedback will greatly encourage our young researchers and contribute to the success of this scientific session. Our respected evaluators are Dr. Praveen Bade sir, founder of Swalife Biotech, and Dr. Vasudha ma'am, Associate Professor at Vignan Institute of Pharmaceutical Technology. Let us welcome our evaluators with warm round of applause. Now, without further delay, let us commence the poster presentation session. Each presenter will have approximately 12 minutes for their presentation. Let us begin with our first presenter. I invite our first presenter, Miss Anik, uh, Anikisha Albert, to kindly present her research work. Is Anikisha Albert there? Okay, so we move further
Okay,
with.
so no, just Ruchira, just put the the name of the presenter in the group as well, so they will connect later. But if they are not available, you can start with the next presenter. That is one thing. The second is that all the presenter will have to share their poster by themselves. We have asked to submit the posters or presentation as a backup. You are going to share your slides from your PC or from your laptop. Okay, so that is one more thing I wanted to add.
So our next presenter is Patil Thorat Nikitasha Bharat. Kindly present poster. We shall now give your very best. Our next presenter, Miss Sandhya, please come forward and begin your presentation. Is Sandhya there? No, right? Okay, so we move forward to Manisha Ranganath Javle. Is Manisha Ranganath Javle there? Next is Mr. Sarang Sunil Mahamuni. Is Mr. Sarang Mahamuni there? No. So we will move further to Mr. Ruben. Ruben, is Mr. Ruben there? Okay. So next is Mr. Germany Danasana. Is there?
Yeah, there is.
Yeah.
Hello, sir. Good morning.
Yeah, good
Yeah. Good
morning.
morning. Uh,
Sir,
please,
I'll try to
please proceed
upload my
further.
slides.
Yeah. Please share your presentation.
Yes, sir. I'll try. Can you see my presentation?
No, we are not visible to you.
Hello. Hello. Can you see my
Can you
presentation
change the
now?
screen?
Hello. Hello. Sir, am I audible?
Yes, you are audible.
Ma'am, can you see my presentation? Hello.
Yes, we can see you. You can start now. Your time starts now.
Okay. So, good morning, respected sirs and madam, and all present over here. I am Germany Johnson from PG Department of Botany, Barampur University. I'm going to present my poster presentation on integrated virtual screening and molecular dynamics approach to identify natural phytochemical as a dual target candidate against the chikungunya virus NSP2 proteins and NSP4 uh, proteins. So, what is chikungunya? Chikungunya is a mosquito-borne viral disease that may be transmitted by some infected mosquitoes like Aedes aegypti and Aedes albopictus. And the genome of this virus is a single-stranded RNA genome, which mainly consists of two open reading frame, that is ORF1 and ORF2. ORF1 consists of four non-structural proteins, that is NSP1, NSP2, NSP3, and NSP4. And ORF2 consists of five structural proteins. The most promising target of this virus is non-structural protein, specifically the NSP2 and NSP4 protein, because of their crucial role in viral replication process. And the most interesting things of this virus is there is no approved antiviral against this virus. So the objective of my work is to assess the binding stability and binding interaction mechanism of the natural compound against the target protein, that is NSP2 and NSP4, and to analyze the conformational dynamics and free energy landscape of a ligand binding, and then to validate the stability and binding mechanism of the lead compounds. So initially, 6,000 compounds were created using the different databases, and then these compounds were screened based on their suitable drug likeness property like ADMT and toxicity. From 6,000 we found 15 hit compounds, and then these 15 hit compounds were subjected to molecular docking against both the target protein, that is NSP2 protease and NSP4 protein, and then the top drug complex are subjected to molecular dynamic simulation study to check their bindings, stability and binding behavior analysis. So the figure 2 represent the binding affinity score of top two compound, that is not diadobertic acid and salicylate against both the target protein, that is NSP2 and NSP4 protein. The figure 3 represents the intermolecular interaction between the phytochemical top phytochemical, that is not diadobertic acid and salicylate against the target protein, that is NSP2 and NSP4. Then the top two complex were subjected to molecular dynamic simulation study to check their binding stability and binding behavior. So we calculate the RMST, RMSF, ROG, and different advanced computational analysis. So RMST stands for root mean square deviation, which mainly calculate the average structural deviation of the protein when in ligand bound position. So you can see that the figure 4 represents the RMST plot for both the NSP2 and NSP4 when in ligand bound position. All the complexes show that the stable RMST plot which suggesting the ligand binding is strong. And then you calculate the RMSF analysis. The RMSF stand for the root mean square deviation, which mainly calculate the flexibility of individual amino acid residue throughout the simulation period. And all the complex show that there is no flexibility in the active site region, which suggesting that the ligand binding is strong when in ligand binding is strong and stable. Then we calculate the principal component and free energy analysis. The principal component analysis is used to calculate the large-scale conformational motion of the protein on a ligand one position. You can see the upper panel represent the PCS scatter plot, the lower panel represent the free energy landscape. You can see the when protein in ligand bound position, there is a more compact PCL cluster seen as compared to the apoprotein, which suggesting that the ligand binding is more stable uh, in all the complexes. The lower plan represents the free energy landscape, which mainly calculate the energetic state of the conformational motion. You can see that the protein in ligand one position, uh, there is more deep energy basins are found that suggesting that the ligand binding is strong. Then we calculate the DCCM, that is a dynamic cross correlation matrix, which mainly calculate the correlated and anti-correlated motion of the protein when in ligand one position. You can see there are two regions, that is one is blue region and red region. Red region represent the positive positive correlated motion, and the blue region represent the negatively correlated motion. So you can see clearly see that when protein in ligand one position, there is more positively correlated motion, which mainly suggesting that the protein and ligand binding is strong. Then finally we calculate the binding free energy analysis using the method MMGBSA to check their to check their strength of interaction between the phytocompounds and the target protein. You can see that in all the complex they show the favorable binding free energy, which suggesting that the ligand binding is strong and stable. So overall, from 6,000 compounds, we found 50 hit compounds from virtual screening. Then these 15 compounds are subjected to molecular docking to check their binding affinity against both the target protein, that is NSP2 and NSP4. And finally, we found the two compounds, that is not dihydrobarotic acid and salitroside as the lead compound among 6,000 compounds. And this suggesting that these two compounds may be a drug candidate for the chikungunya virus. Thank you.
Thank you, Germany Danasa.
Hello. Hello, can you hear me?
Yes, yes, sir. I can hear.
Okay, so there is one question from my side. What is your hypothesis?
Sir, my hypothesis is to inhibit the target protein, that is NSP2 and NSP4 of the chikungunya virus.
Okay.
Yes, sir.
And how are you inhibiting them? By small molecules?
Yes, yes, sir. By small molecule like natural compounds, or we can design some synthetic compound also to inhibit this
O-
target protein of the chikungunya virus.
So you are synthesizing the molecule.
Yes, sir. We can
Okay.
synthesize.
Okay. Okay. Thank
We
you.
can
Thank you.
synthesize. We can design. We can also design natural peptide. We can also design chemical compounds, synthetic
Okay. So
compounds,
where
or
are you
another.
in this project? You already started the project, or you are
Yes,
just
sir.
actually
Already
developing the
started.
hypothesis?
Already
Already
started,
start.
sir. Yes, sir.
Oh,
Yes.
okay. 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 Thank you.
Thank you, sir. Thank you so much.
Okay, now our next presenter will be Manisha Zavre. I request Manisha Zavre to present her work.
Ruchira, you need to remove other people from the stage and add Manisha on the stage.
Yes, sir, removed. Manisha, you can start now your work. Manisha, am I audible to you?
Hello. Manisha is not audible.
Manisha, can you hear us?
Hello, Ruchira. If Manisha cannot do, then we can take the next presenter.
So actually, next presenter is not over here.
So, who is the next presenter?
Next presenters are like Anishika Albert, and she is not over here. Nikitisha Bharat, Sandhya.
Put their name in the group so they will join.
Yes, sir.
Likita, share the names of the presenter, oral poster presenter in the group. Hello, can you hear me?
Yes, sir, shared.
Okay. I tell them the slot number as well, and I also ask them if they can join or not. We already shared the StreamYard link. Can you share the StreamYard link in the group as well?
Yes, sir, we can share StreamYard link. Sandhya, are you here now? Sandhya, am I audible to you? Sandhya, am I audible to you?
Hello, Sandhya.
Sandhya, are we audible to you?
Hello. Hello.
Sandhya, are we audible to you?
Ma'am, it's audible, ma'am.
Sorry?
Hello.
Yes, Sandhya, please present your work.
Okay, ma'am. Ma'am, am I audible, ma'am? Hello, ma'am.
Yes, you are audible. Yes, you are audible to us. Please share your screen.
Ma'am, I shared, ma'am.
Sandhya, we can't see your work.
Ma'am, 1 second.
Sandhya, you can start now.
Hello, ma'am.
Yes, you can start now.
Hello. Good morning to one and all. நான் பேசுறது எனக்கு கேக்குது.
Sandhya, you are audible. Start with your poster presentation.
Sandhya, your time has already started.
Good morning to one and all. My name is Sandhya Muthu, research scholar from Department of Pharmaceutics, SRM College of Pharmacy. My research topic was on QBD-based development of silestazole-loaded transdermal nanoemergel for the management of Raynaud's phenomenon. Raynaud's phenomenon is a chronic peripheral microvascular which affects the episodic vasospasm of the digital arteries, which leads to the reduced blood flow in response to the cold exposure or emotional stress. So, conventional therapy often produces systemic adverse effects and which undergoes a first pass metabolism. So, I have chosen the transdermal drug delivery approaches to alternate the enabling sustained drug release to improve the patient compliance and avoid gastrointestinal degradation. And also in the transdermal
Sandhya, you only have more 5 minutes to present your work.
What happened, Sandhya?
Sandhya, we are not audible.
hello ma'am.
Yes, now you are audible.
Okay ma'am. And then
Please
I
start
have
your work.
chosen.
Continue, please.
Okay ma'am. So in transdermal approaches, I have chosen nano ml gel, which gives the nano sized droplets and enhance the skin permeation with increased solubility of the drug, and it also targeted the peripheral microvascularization of the. Okay, ma'am. So the research gap was found to be the current oral therapies associated with systemic adverse effects, and only limited transdermal approaches were available for Raynaud's phenomenon. Few studies have been explored that QBD based optimization for the vasodilatory effects. So there is a need for stable transdermal nanoemer gel, which will be capable for enhancing the peripheral microvascular perfusion. So the rationale behind the choosing of celestosol drug is it is a selective phosphodiesterase 3 inhibitor which has four main mechanisms. It has a vasodilatory effect, antiplatelet effect, improved peripheral.
Sandhya, you only have 2 minutes to present.
Ruchira, can you take next?
Yes, sir. Sandhya, please stop your work. We will go further with Sarang Mahamuni. Sarang Mahamuni, are you there?
Manish,
Yes, ma'am.
Yes, Sarang, please start your work. Your time starts now. Please share your
Okay.
work.
Is this visible, ma'am? Yes, ma'am. Okay. Good morning, everyone. Today I'm going to share the review on the drug discovery and the development using the network pharmacology, which is an emerging system biological approach or biology approach. In this particular idea of the discovery of the drug is emerging one, that is the drug discovery has evolved from the conventional method, that is one drug to one target. We have interact the drug and we got idea about there is a interaction and the activities there. But this strategy is the old strategy nowadays for drug repurposing, the system-based approach that recognize the complexity of the biological network. That is, number of activities will be there for the one drug is there. So we can easily understood the particular drug interaction with the different genes of the targets by using this network pharmacology. So we involve a network of the different diseases. So the network pharmacology integrate the pharmacology, then bioinformatics system and the biology and the computational sciences by identifying this particular interaction of the among these bioactive compounds, different molecules, different drug molecule derives from the plant itself, then signaling pathway and the diseases. This study highlights the application of the network pharmacology in
hello.
the discovery. Yes, am I audible, sir?
So,
Am I audible?
Yes, you are audible, Sarang.
Yes, this study highlights the application of this network pharmacology and the discovering associated with this particular genes, and by using the gene card, OMMI, and the DGEN net. That is, these are the online softwares and the informations available. For example, OMMI means the online Mendelian inherent in a man, that is the human gene. We can easily find out the gene associated with the diseases. The common target between that diseases we can identify it using the van analysis and the user for the construction of the protein-protein interaction, that is PPI. Means we can first of all select the ligand which is come from the natural sources also or synthetic sources also, and we map all the genes of a particular drug with a proteins genes of that targets, and we can easily get idea about a network of that particular drug interaction through the network pharmacology. Or cytospace is um, one of the site where we can easily interact this network. Then functional enrichment analysis we can done by the gene ontology, which is called as a GO, and the site cake pathway or the keynotes encyclopedia gene validation, which is that genomes, that is called as cake, and where the perform the elucidated whatever the biological processes and the signaling pathway which involved in that we can easily identify by this network pharmacology without using the platform of the without using the platform of the animal study. We earlier doing all this approach in a in silico manner in a computer itself. Okay, so it is easy to identify the different genes involved in these diseases by the single drug also. So this integrate the analysis of identify the several key targets associated with the specific. For example, there is a case is that inflammatory studies during the docking. We can identify this, that is oxidative stresses, apoptosis, immune regulation. We can identify, demonstrate the multiple target potential natural compounds also, and strong molecular interaction we can done during this docking. Finally, after we can get idea about the drug is have the proper interaction with that genes, and we get the pharmacological activity in silico. So these findings suggested that a network pharmacology is a valid platform for the accelerating the discovery and reducing the development time also, because what is happened, whatever the traditional route is there, finding out the discovery of the drug required lots of a time, and because of this network pharmacology within a within a 2 hours also we can get idea about which drug is multiple target with the different genes of that particular diseases, so that it is a less time consuming process than that of the traditional method. So it is cost effective also, and facilitating the identification of the safer and more effective multi-target therapy also. So at a one time we can easily get interaction with the different or multiple targets also. This integrated computational strategy provide a scientific base for the further experimental validation also. So before going to do the any experimental work on any animal, ethically we can really get idea about it is feasible to do the work. If it is feasible to do the work on in silico, you can easily apply this particularly approach for the animal also. So it is a validation process, supports the development of the novel pharmaceuticals for the treatment of the complex disease also. So this is the introduction related to the development of the drug by using the network pharmacology approach. So in this case, we can go the step by step. For particularly one step is, or first step is that data collection, or it is called as a data mining also. So first of all we collect the data of the particular disease and genes related to the disease. Okay, so this is the first step. And then we also collect the drugs by using the different types of the sites are there. Duke, that is Dr. Duke site for the identification of the plants and their different types of the molecules which involve in the active molecules. We can find out easily. We can find out the PubMed drugs from the PubMed also, PubChem also, and we can get idea about traditional Chinese medicine system pharmacology, that is TCMS site. So different sites are there. From that we collect the data of the drugs. There are different types of the chemicals involved in the drug, and chemical involved in that drug development, which is come from the natural origin also. And we screen this afterwards in a next stage. So first of all, we collect the genes of the particular disease, we collect the targets, and then we go with the target prediction. So using the computational logarithmic to the screening method, that is called as the first of all, we get whatever the information we apply for the compound screening. So active compound selected here, herbal drug from the TCMS, that is the traditional Chinese medicine, and PubChem and the Swiss. We go with the target prediction. The compound targeted are predicted using the Swiss target prediction site. So Swiss target prediction and the pharma mapper is also. So these are the sites are there. We can map the target and we get a common genes which is involved in that particular plant or the drug or the different ligands. And then we move towards the network construction. We construct the network, that is we construct the network by using whatever the we collected data, we whatever the common genes are there, and we interact drug disease target network. We form the target, whatever is the interaction is given in the form of the target common genes we collected, and we get a nodes and edges in this case. And whatever the figure is given in this network pharmacology, these nodes we can easily get green color and red color nodes are there, and whatever the edges are there, which is given in the form of lines. So that edges and lines and the compounds we can easily identify by the network in that. Then we move toward the network analysis, that is the topical analysis with the perform to identify the target and the hub protein. So according to that, whatever we got targets in that image, we collect the data and we can get idea about which is the common genes involved in that disease target, and we select this disease for the further further studies on animal also. Then we enrichment of the animal. So the in between that enrichment, the pathway or in the cake pathway, already we discussed that gene ontology and the cake pathway. So this gene ontology can cake pathway which involve in that we find out the pathway of the disease where these particular drugs are involved in that. Different types of such tools are available online. Tool String is the one of the site. It is very important site, and Metascape site is also there. Then DAVID, this site is also there, where we can find out the gene ontology in this. Then molecular docking. Finally, we do the molecular docking, that is to simulate the binding of the compound to the predicted target. So whatever the target we selected from that, we go for with that proper interaction is there or not by using the molecular modeling. So simulate the binding compound to predicted target. We can use online AutoCAD also, Vina, then YASARA, SIRA also, and confirm the binding of this particular drug interaction. So I hope you can get idea about this molecular docking image is given, that is how intrinsically or the hydrogen bonding interaction we can find out in that drug likeness. We also find out this by using the softwares, and different types of the online platforms are available that are there. So we can also understood the enrichment of analysis, which is given, that is the inflammatory responses are given, immune responses, apoptosis process responses, oxidative stress given the pathway by using gene ontology biology. And cake pathway also is given, which is specific type of the apoptosis, MAPK signaling pathway involved in TNF pathway in inflammation. So we can easily identify this particular type of the interaction also in virtually, instead of by doing all these things on an animal. So there are the total 126 active compounds we can also taken into this drug at a one time, and targets maybe more than 200 also we can at a time get idea about the interaction, and from that common targets we go for the further studies by using the specific type of the disease targeted on this. So I hope you all understood about the comprehensive overview of the drug discovery and the development by using the network pharmacology as an emerging approach. There are lots of the papers are available in within a short period of time. We can do the work for the specific diseases and the targets also. So I hope you are understood. So can I stop here?
Yes, Sarah. Now we will go with one question to you. What
Yes.
makes the identified compounds promising candidates for further drug development?
We can I already we collect the data for the drug itself. We get idea about the which type of the target will be easily interacted for that activity. So already get idea about the drug is there, but sometime what will be happen, this particular type of the software or the site will use idea about drug likeness also by using the Lipinski rule, and drug likeness is not there, then we cannot use further analysis.
So next question from Vila sir is that what do you mean by systems biology approach? Please answer to them.
So system biology, so what approach is nothing but what systematically from different types of systems available for the network pharmacology and biological approach, we can combine itself, and because of that we can get idea in a proper manner. So if it's a system is there, that system contain different types of the softwares, different types of the online platforms for the targeting the drugs, genes, finding out the genes, mining of the predicted targets and all. So because of that, we call it the system biology.
Okay, thank you, Saran.
Okay, thank you.
So we move further with Manisha Zavre. Manisha Zavre, are you ready with your presentation? Manisha, are you audible? Manisha, are you audible to us? Hello, Manisha. Manisha, please start with your work. Your time has already been started. Manisha, your poster is already been shown over here. Please start your presentation. Manisha, please start your work. Manisha, you only have more 7 minutes to present your work. Manisha, please start your work. Manisha, we are not audible to you. Please start your work. Manisha, we will stop now. Time is over with you. Is other any poster presentation present over here?
Good afternoon, everyone. Conclude our presentation. Now we shall move towards the second part of lecture. On behalf of the organizing committee, we sincerely congratulate all poster presenters for sharing your valuable scientific work. Now, it is my honorable to introduce our next dignitary speaker, Dr. Dr. Nilesh Narayan Prasad is globally recognized physician educator, researcher, author, and advocate of sustainable healthcare. Throughout his, throughout his career, he has worked in medical education, healthcare leadership, innovation, and sustainable development. His commitment is improving global health and promoting collaboration has him recognition across to the international scientific development globe. In today's rapidly evolving world, sustainable development has become as essential responsibility for healthcare professionals, researchers, educators, and policymakers. We are privileged to hear Dr. Prasad to speak how science and and contribute. So, everyone please join me. Welcome Dr. Nilesh Narayan Prasad. Sir, the stage is all yours. Thank you.
Good afternoon. Are you listening to me clearly?
Yes, sir. Your voice is clear.
Okay. Welcome. May I start? May I proceed?
Yes, sir, you can proceed now.
Okay, thank you. Honorable President of this organizing committee, Chair of these programs, Excellencies, distinguished personalities, galaxy of scholars, chain of research scholars, academicians, scientists, professors, industry experts, students, and team members. Really, it's the good moment, feeling happiness, pleasure. Thankful to the organizer to give opportunity to me to represent myself as a speaker, as a panelist, to speak on sustainable development goals. Sustainable development goals, not a new things. I will make explanations. I will go related with the SDG 3 also, that it refers to good health and well-being. At first, I will give the introductions of these SDG programs. I have been working as a co-project director and a speaker. I have made the association with different organizations having the association with United Nations. Sustainable Development Goals launched by the United Nations in 2015. It is 15-year programs that will end in 2030. Please make confirmation, are you listening clearly or not? And at the beginning of this program, I was finding that there is a technical problem from your side. So please, I have requested, please make the confirmation like that, are you listening my voice clearly or not?
Yes, sir, we are audible to you.
Okay, thank you. Thankful. So my focus, emphasis should be on the Sustainable Development Goals launched by the United Nations in 2015. It's the 15-year programs that will end in the 2030. In these 15 year programs, we have to accomplish the objective and mission of the United Nations. I expect I have been passing through the 2026. It means 11 years have already passed, and now we have only the 4 years for the accomplishment of the objective of United Nations. In these 4 years, how can you approach this? How can you accomplish this? There are a chain of the hindrances, troubles, and impediments in the way of the achievement and accomplishment of the objective of United Nations. It's not the new things. I am not talking only the Sustainable Development Goals programs. Just I will take the 2 and 3 minutes for saying about that basics, introduction of it, history of it, so that the persons who are not having the association with United Nations and Sustainable Development Goals plans, they will be also aware about that. Why we have to be participate in the Sustainable Development Goals plans and how it has been launched and what is the motives and objectives of this Sustainable Development Goals plans and why we are making discussion on the same issue, same topics, why that gets the priority not only by the organizers and associations, by the United Nations and the different agencies having the association with the United Nations will prefer to talk, make discussion, make the webinar and organizing the events on the Sustainable Development Goals plan. It is not only for a particular organization. That mission is associated with that is the United Nations, having the association with the 193 countries across the world. That's why it has got the priority by the government body, by on the international platforms, by the agency of the international United Nations also. Before the commencement of the Sustainable Development Goals plans, there was also the existence of the Millennium Goals plan. Millennium Goals plan was launched in the 2000. It was also 15 years programs that was ended in the 2015. That was also 15 years programs. They started in the 2000 and ended in the 2015. But what was the difference? Why it differs from the SDG program? Why we have to launch the SDG program? That question rises. Before the commencement of the SDG program, there was the existence of the Millennium Development Goals plan, MDG that is. That was also 15 years program, but there were only the 8 goals. There, that is the point is to be noted. In Millennium Development Goals plan, there were only the 8 goals. 8 goals mainly focusing, emphasizing upon the elimination of the poverty. Elimination of poverty, removal of poverty only. And that was the development in terms of the economical development, social equality also. After that, there was a conference in the Rio, Brazil in 2012. As a result of this conference, there was the commencement of this SDG program. That conference was launched in 2000, was held in the 2012. But this SDG program has been launched in 2015. That is also the part of remarkable. That is the to be making the discussion on the topics. Why there was the delay of the 3 years. Actually, that was the 20 years program. That is the 20 plus events that is the held in the 20 and for the 2 stage programs. Launch that was the conference held in the 2012, but after that conference, it was taken decisions by the members of the United Nations. We have to launch the next goal, next mission after the completion of this Millennium Goals. That's why a new mission, that is the project was launched by United Nations in 2015. That is the Sustainable Development Goals. That is the result consequences of the conference held in the 2012 in the Brazil. A number of the members of the United Nations were participated in that programs in the conference, and unanimously a decision was taken for the launch of the Sustainable Development Goals Plan, and that was launched in the 2015, having the 15-year program, and that will end in the 2030. We are passing through 2026. It means now we have only the 4 years for the completion and for the success of this plan. Sustainable Development Goals Plan that was launched in 2015, and now these Sustainable Development Goals have the 17 goals. In the Millennium Development Goals, there were only the 8 goals, and now the Sustainable Development Goals, we have the 17 goals. That was the difference. One main difference that was in the Millennium Development Goals Plan, that was also for 15 years programs, started in 2000, ended in the 2015. But only the developing country, the development of the developing countries were put under consideration for that programs. Developed country was not that is the put under consideration for giving the priority. Only the developing countries were given the priority. That was in the MDG, that is the Millennium Development Goals Plan. But in this SDG, Sustainable Development Goals Plan, what is the priority? One, not only the developing country, developed country were also put under consideration. Developed country were also put under consideration. That was the basic difference that must be kept in mind, that in the Millennium Development Goals, only developing countries were put under consideration for the development plans, and the program mission was for the developing countries. But in the SDG programs, not only the developing country, developed countries were also put under consideration. With this program, this sustainable initiative is not only for the developing country, that is for the developed country also. The participation of the developed country in the execution of the missions and for the completion of the objective, that is remarkable point for Sustainable Development Goals. There are 17 goals. 17 goals, that is the among 17 goals, some have the social foundation, some have the economical foundation, and some have the environmental foundations. For this and last 2 are, that is the under 17 goals, that is the first starting from the no poverty, that is the first SDG goal. No poverty, zero hunger. Zero hunger is the second. Good health and well-being. Good health and well-being, that is the 3. Quality education, goal number 4. Gender equality, that is the SDG 5. SDG 6, that refers to the clean water and sanitation. SDG from 1 to 6, that it comes under the ambit of social foundation. As per the guidelines of the United Nations, these 6 come under the ambit of social foundation. And after that, starting from the 7 refers to the affordable and clean energy. 8 refers to decent work and economic growth. 9th is for the industry, innovation and infrastructure. 10th, that is the reduced in equality. 11th refers to sustainable cities and communities. 12th refers to responsible consumption and productions. So starting from the 7 and up to the 12, these come under the ambit of economic and infrastructure foundations. I will correlate it just after giving the completion of this step first lecture. I will deliver the lecture how these are interlinked with it and why we have assembled here for making the discussion and for making the question answer and interactions. So all will be discussed step by step. Just I am giving. So these are up to the 12. And now it starts for the environment. In environment foundations. Starting from 13. 13 refers to quality, that is the climate action. 13 for the climate section refers to the environment. Climate action refers to environment. After that, 14, life below water. 15, life on land. These come under the ambit of environment. After that, 16 and 17, peace, justice, and strong institutions refers to 16. And partnership for the goals, that refers to 17. These two are for the proper smooth functioning, for the proper executions, for the implementations in a smoothly, for in proper way. So these are the 17 goals of, just I have explained, I have put the name in sequence. One thing, these are in sequence, and just I have given the ideas what are the motives of the United Nations. First 1 to 6 come under the social responsibility foundations. After that 7 to that the 12 come under the economic and the infrastructure. After that we study about the environmental impact, environmental missions, environmental foundation, sustainable environment. After that we have to organize it peacefully. We have to make the collaborations, partnership with chain of the institutions, chain of the associations, not only the national level, at the international level also. We have to establish it at international level also. So these 17 goals are sustainable development goals launched by United Nations. United Nations, it's the project of United Nations, so it's gets priority by the different associations and organizations. It has a long impactful effect also. Just I will explain it, why we have to carry the sustainable development goals plans, why we have to work on that, what will be the advantages, why we have to adopt the sustainability concept of the United Nations. So all will be discussed. It has the great impact, long-term impact. We have to follow it for the welfare of the country, for the welfare of the society, for welfare at global level. So we have to adopt it. As far as so health, that is the program is also associated with the health and health programs, and that is the good health and well-being. That is the SDG 3, comes under the ambit of the Sustainable Development Goals Plan in SDG 3. But SDG 3 cannot be achieved, accomplished without the achievement of the SDG 1 and 2. If there would be poverty, if there would be hunger, if you are not getting the food, if you are not feeling the energetic, if you are not taking the nutritious foods, balanced food, and if there would be the lack of the money, liquidity to purchase the food stuffs, for taking the food and for getting the energy, is it possible to be the part of the good health and well-being? Impossible. We cannot carry, you cannot fulfill the concept of SDG 3. Good health and well-being completely depend upon the first two goals. If there would be poverty, poverty means if you are not able to afford the foodstuffs which are needed for the good health and well-being, if you are not taking the balanced food, Not I am talking the balanced food. Balanced food is a very high affordable food stuffs. Just I am talking about the basic needs. If you are not taking the basic foods needed for our body, for the well health, nutritious food at the basic levels. I am not talking about the high level and therefore a balanced food like the having the nutritious food, having the sufficient amount of the calories. I am not talking about that. Below the calorie, we actually, as per the United Nations data, more than the 700 million people, 700 million people are living the pivot, that is the below the poverty line, are facing the poverty. It means poverty means if the person is not getting $2.15 per day, its source of income is below this $2.15 per day. It means they will be considered as a under the ambit of poverty. It means below the poverty line. And we have to eliminate it. We have to remove it. How can you achieve for that? For that, we have to take decisions not only at the national level. We have to organize the awareness programs. We have to launch the schemes of the government at international level. We have to organize the events, making the collaborations at different international platforms. And developed country has to take initiative for that. They have to assist the developing countries and the Banana Republic poor country by giving the financial assistance and by making the assistance in terms of the providing the resources. So developed countries have significant role for the eliminations of the poverty at international level, at global level. So that is the part of the first sustainable goal. We have only the 1 hour, so I will take only the glimpse of it. In the future, if I will get the opportunity, I will explain it in details also. That is the continuous process, not only restricted for this program only. So in these programs, we will put the highlighted points only. It is a continuous process. Sustainable development goals cannot be restricted in 1 hour and not restricted to a particular organizations. It is a global level missions, global level objectives. We have to make the partnership collaborations with international platforms for the accomplishment of sustainable development goals. So now second, that is the after the no poverty and zero hunger. It is impossible. Zero hunger means every one person, not a single person, will sleep without taking the food. Each and every person should be provided foodstuffs, what they need. And is it possible? For that we have to launch mission. That's why we have assembled here to talk about the sustainable development goals. And if you are not able to accomplish that second goal, that is the zero hunger, then how can you accept, how can you expect, how can you achieve, how can you fulfill the objective of SDG 3, good health and well-being? Good health and well-being is completely dependent upon what you take, where you live, and what is your environment, where you survive, climate conditions, air, what you take, water, what you consume, and foodstuffs, what are taken by you, by us. That is the primary requirement for the determination of good health and well-being. So nutritious food, climate, having fresh air, fresh water, fresh climatic conditions, temperature appropriate for the survival, and balanced ecosystems. all are needed for the good health. Yeah. Yeah, are you listening now?
Yes, sir.
Okay, thank you. Actually, there was the shifting in the Wi-Fi. That's why there was disturbance and disconnection. So sorry for that. Now Wi-Fi has been changed now completely.
Yes, sir.
Now
No
it is
problem,
clear.
sir.
Okay. So now, so we are talking about the good health and well-being. Just I have said, I have used the terms like that, clean water. We have also used the terms like the clean, after that, clean water, climatic conditions, suitable climatic conditions, fresh air. We have talked about the temperature. We have also talked about the balanced ecosystem and foodstuffs needed for our body. But is it possible to get all what we expect in the same way we will consume it? We will get it? Not. If you talk about the environment, if you talk about the climatic action, that refers to SDG 30, that is for the climate action. If you talk about the clean water, that refers to SDG 6. If you talk about the energy, affordable energy, it refers to the SDG 7. If you talk about the life below water, ecosystem, balance in the ecosystem in the food chain, that refers to SDG 14 and SDG 15, life below water, SDG 14, life on land, SDG 15. So that is completely related to the SDG 14 and SDG 15. Actually, we are talking about the SDG 3, good health and well-being. Why we are discussing about the SDG 14, 15, 13, 6, 7? Oh, all are interlinked. If there would be disturbance in environment and climatic condition, we can't expect the execution of the concept of good health and well-being. For the proper execution of the concept of the good health and well-being, there must be proper climate action. Climate action, SDG 30. Clean water and sanitation, SDG 6. Affordable and clean energy, SDG 7. Life below water, proper ecosystems, food chain balance, balance ecosystem, SDG 14, 15. So we have to adopt the concept of United Nations in terms of the proper execution of the all 17 SDG programs. SDG, all 17 goals are interlinked. Without the execution and the proper execution of the SDG 1 and SDG 2, we cannot expect SDG 3. And without the execution of all the SDG that I have explained here, you cannot expect the SDG 3 also. And now, if you are not taking the sufficient amount of the energy needed, calories needed, foodstuffs, there would be problems, health issues. And sound body has the sound mind. If there would be no sound body, it is the beyond the expectation. If you expect we will have the sound mind, it is beyond the expectations. We cannot get the sound mind without sound body. If you are not healthy, mind would be not, you're not at up to the mark needed for the execution of your plan in the proper way. So we need good health, sound body, sound mind. So without the sound mind, your efficiency, productivity will be not up to the mark. If you are talking about the efficiency and the productivity, innovations, that refers to SDG 9. All are interlinked. That refers to SDG 9. If you are talking about the productivity and a result, consequently, we will contribute in the growth of the economy. Economical growth. If you are talking about the economical growth, that refers to SDG 8. Remarkably, we are talking about the good health. And in second, on the other hand, we are talking about the economical growth. Economical growth, that is SDG 8. That refers to SDG 8. So in one hand, we are talking about the good health and well-being, SDG 3. On the second hand, on the other side, we are talking about SDG 8, economical growth. And if there would be the proper growth, economical growth, that will impact upon the economical growth of the country. Economical growth of the GDP. If our country will rise, economy will rise, it will have the impact upon the global economy also. So if we want global economic development, rise in the standard of living, rise in the per capita income, elimination of the poverty, we have to adopt the schemes, concept, ideology, different missions launched by the United Nations, and we have to collaborate at the national and international levels, organize the events, series of programs to make the aware. Large sections of the people are living in the remote areas. We are not talking only about the India. If we have to talk about the sustainable development goals, we have to talk at the international platforms. We have to talk globally. We have to talk about the different organizations and associations across the world at international platforms. Large sections of the persons, large sections of the people, citizens, not only citizens with having the association with 193 countries, being the members of the United Nations. They are not aware. Government, our India, if you consider about the India, our government has launched a chain of schemes, programs, but the persons living in the remote areas, persons living in the villages are not aware, unaware. They are not aware about the schemes and the programs launched by the government. But in some cases, as per the data released by the Niti Aayog, Government of India, of index for the SDG 7. SDG 7 refers to the energy, affordable energy, clean and affordable energy, that is the SDG 7. That is the best performing goal out of the 17. Remarkable point. That is the SDG 7, clean and affordable energy. That is the best performing goal out of all 17 goals. And as far as the score is concerned by the Niti Aayog of Government of India, its score is 96 out of 100. That is remarkable. That is to be noticed, that will be put in on record. 96 out of 100, being on the driving seat, in leading role as far as the all sustainable goals are concerned. Out of all 17 goals, on driving seat, that is the SDG 7. I have been working on that. I am also the speaker, and that is the partner for different organizations on the SDG 7 also. So SDG 7. So, and overall, if you are considered about the overall performance, that is the score is only 68%, approximately 68%, 68 point something like that. So why we lack the 32%? If there would be the score 68%, so it means we are We lack the rest 32%. So what are the loopholes? What are the hindrances, impediments in the way of the achievement of the goals, target? That is lack of the awareness programs. A large section of the people are living in the remote areas. They are unable to the schemes. How can they avail the schemes and programs launched by the governments? They don't aware about that. So we have to organize the series of programs to make aware the persons who are living in remote areas and villages. They should be put in, they should be brought in the streamline of the government programs and schemes. They should be considered, they should be brought in the streamline. They should be aware, they should be aware by organizing a series of programs, webinars, symposiums, sometimes a particular NGO and particular workers, volunteers of the United Nations should visit the remote areas and they should organize the programs to make the aware. And not only this, if they need the resources should be provided also by the different organization, NGO, civil society policy forums of United Nations, World Bank, etc. So in this way, we can fulfill the required efforts, course of actions to fulfill the gap of 32%. For the accomplishment of the data of 100%. For the accomplishment of the objective of United Nations in this way. So, just I have explained, all are interlinked. Good health and well-being can't be considered without the execution of all other goals in proper way. In proper way, it means we have to execute it, we have to make aware. Actually, a large section of people are not aware about the SDG programs. A large section of the people don't know what are the United Nations, what are the SDG, and how and what is the mission of it, how these are advantageous, beneficial to us, why we will be the participant in these programs, why we will act as a volunteers, what will be benefits. A large section of people will think only about the financial assistance, about the financial advantages also. Not only this, if we work on the SDG programs, in one hand, we will have the recognition of the volunteer of United Nations. In second hand, we will get the recognitions at international platforms. After that, we will have the association with the 193 countries. We will be on the international platforms. And now, just I have explained about all are interlinked. So now question rises in a large section of the people will think about that. How can we the participant? How can we launch the programs, sustainable development goals programs? How can you proceed for the accomplishment of the rest targets? Actually, that is the 15 years programs. 11 years already passed. Now we have the only 4 years. So it's the great challenge before us, not before us. That great challenges before the government, before the different agencies of the United Nations, before the developed countries also. They are also facing challenges. Government body are facing challenges in this short period of time. And 4 years we have to accomplish the target. We have to achieve the target. How it will be possible? As per the data, 4 US trillion are needed per year only for the developing countries for the smooth functioning of these SDG programs launched by the United Nations. That is, it's a big amount, big investment. For that, developed countries, being the economical power, should take the initiative, should make the contributions for the banana republics, for the developing countries, for the poor countries, for making the awareness programs in the remote areas, in the villages. They should take the initiative, making the contributions like the financial funding. Not only the developed country. And now just I am taking the 2 and 3 minutes by saying about the procedures. How can you approach to the United Nations? Actually, anyone who is interested for making the contributions in this good, that is the big project of United Nations, what is the process? Just I am saying in the 2 and 3 minutes, they can contact me, they can make the association with us also. Just after giving these ideas about that, you have to approach to the portal of the United Nations. There is the link for making the registration, partnership and collaborations. There at first we have to register your organizations. On the behalf of that organization, you have to represent yourself as a volunteers. There you have to make the registration on the portal. After making registration, giving the all details about that and why you are making the, how can you approach for making the contributions, what is your objective, all you have to mention on the portal. What will be the instructions, we have to fulfill it. After making that is the registration for that, after that you will get the project ID. Project ID will be issued by the United Nations. That is important. And now under that project ID, we have to carry a series of programs. You have to carry a series of programs under that project ID, and that programs, that events should be circulated on the social media, should be shared on the social media, and that is noticed by the United Nations. If under that particular project ID that is issued by the United Nations, if you carry a series of programs, and if that will be circulated and shared on the social media, that is noticed by the United Nations, that must be kept in mind. And not only you have to carry the programs at national level and your state level, that you have to carry the programs at international level also. That even you have to make the collaborations, we have to make the partnership with international association and international platforms, different bodies. And that will be noticed. And now if you need the fund for carrying your tasks at global level, there is also the provisions of funding from the United Nations. But you have to, at first we have to take initiative. That must be noticed by the United Nations if you approach for the financial assistance. If you need the fund for global level events, for making the collaboration, organizing the events at international platforms, if you need the fund, there is also the provisions of funding from the United Nations. You can apply for the funding and you will get the funding, financial assistance from the United Nations also. That is the way, in this way, we can make the contribution to United Nations, these missions, these sustainable goals plans. And not these sustainable goal plans, not only for your sustainable environment, for social work, for economical development, for sustainable environment, for your recognitions, for your recognition at national platform, for the recognition at international platform, to be the recognized as the United Nations volunteer. You will make the association with 193 countries. 193 countries being the member of the United Nations will be in contact with you. We will have the approach to 193 countries. You will be on the international figure. You will be the volunteer not only for the United Nations. We will be the volunteers of the 193 countries. We will have the association for that. That is the great recognitions for your effort, for your course of actions. In one hand, we will get the recognitions. On second hand, we will be on the international platforms. If needed, you will get the financial assistance also for the launch of your missions under the ambit of, under the umbrella of United Nations. That is, you have to make the partnership. Today, Sunday, I have been working on this United Nations Sustainable Goal Plans also, that will be live on the YouTube also. If anyone wanted to get the certificate, that will be the world record also. That is the marathon. That means that will be the continuous programs starting from the 3 p.m. today, and that will end tomorrow at 5 p.m. That means more than the 26 hours it will be carried out continuously. And that is under the Project Active United Nations. So in brief, we can say it's the Sustainable Development Goals Plan. All are linked with, if you want to be part of the good health and well-being SDG 3, we have to take actions for climate action SDG 3. Climate action refers to ecosystem, balanced ecosystem. Balanced ecosystem means balanced food chain, free from the contaminations, free from the pollutions. Nowadays, we are living in the environment, air is completely polluted in our areas. Not only I am talking about the India, at global level also. There is the presence of the contaminated pollutants in the environment, in the air. We are taking the contaminated water, polluted air, polluted environment, contaminated water. In this scenario, if you consider we can fulfill the concept of good health and well-being, it is impossible, can't be achieved. So we have to take the climate actions. Climate action, it means we have to reduce the greenhouse, we have to reduce the chlorofluorocarbon gas, pollutants in the air, act upon the greenhouse effect also. You have to reduce the possibility of the destruction of the ozone layer. Ozone layer is being destructed by the emission of the harmful gases, chlorofluorocarbon gases, etc., responsible for the destructions of the ozone layers. We have to reduce the possibility of the harmful gases, possibility of the emission of the harmful gases in the environment, in the air, in the atmosphere. There is also the possibility of the acid rain. Acid rain, harmful gases which are released in the environment, they react with the sunlight, in the presence of the sunlight with the air molecules, and then they will be converted into the acid, and that acid, if come in the droplets, in the water molecules, with the rain, that will create, that will form what is acid, and that is called acid rain. Acid being mixed with rain will cause the acid rain. So we are facing the problem of acid rain, sulfuric acid, by the release of the sulfur dioxide gas that will be mixed with the hydrogen water molecules in the presence of sunlight, and that will create the sulfuric acid, and that sulfuric acid will come in contact with the rain, and that will come on the surface of the earth. That is called the acid rain. The formation of the acid in the, but why we don't feel acid? Sulfuric acid is highly harmful acid, and but we can't feel the immediate effect. Why? Now it is in very less amount, and rain water is in large amount, and that is mixed with the rainwater, so it has the very less impact on the surface of the earth, on the life of the flora and fauna. On the life of the flora and fauna, it has the very less impact, but its concentration is increased gradually, rapidly. Now we can say, in course of time, in the future, there would be the percentage of this concentrated hydrosulfuric acid in more in amount, and that will have the remarkable harmful effect on the life of flora and fauna. So we have to be careful about that in present time. Carbonic acid, a large number of the industries in the Agra region have been closed. What was the reason? Taj Mahal, that is made up of carbonic acid, that is the marble, and by the effect of the carbonic acid that was being destructed, carbonic acid, by the release of the different harmful gases and carbon dioxide from the industries, there was the formation of the carbonic acid by mixing with the rainwater, and that was coming in contact with the marble by which the Taj Mahal was made of, and that Taj Mahal was being destructed. That's why a large number of the industries were closed, shifted to other area. Not only closed only, they were shifted. So that was the course of actions taken by the government. But not only government, we have to take initiative for that, for the safeguard of the life of the flora and fauna. That is, so all are interlinked. If you take initiative in this scenario, we have to launch the programs. Remember, every action counts. Every action counts. Let's multiply our impact. We can achieve the goal if you are determined. Remember it. What we do today, we will get tomorrow. We have to transfer the legacy of hope. We have to transfer, we have to carry the legacy of hope to generation to come. So we have to be highly careful. We have to execute it in proper way. We have to transfer the legacy of hope to generation to come. We have to be accountable. We have to be responsible for the resources and for the consequences which will be faced by the generation to come. We are accountable for that. Next generation will ask questions. We have to give answers. And for that, we have to determine for the proper execution of the mission and objectives of United Nations. How it will be executed in proper way. Only thinking about the good health and well-being has not the fruitful will have not the fruitful result. Think about the all SDG, because your good health and well-being are completely dependent upon the other factors. Other factors, just I have explained, it's a very short period programs. So just I have explained it in brief. It's like the scientific approach. I had given ideas about the sulfuric acid, about the carbonic acid, how it will have the impact, adverse impact upon the Taj Mahal. Just I have explained. It has the scientific approach. Here I am not going to explain the scientific approach. Whenever I will get the opportunity, I will also explain the scientific approach also. It has the harmful effect on the life of the flora and fauna also. Human beings included in this. So without this, we cannot expect the good health and well-being. So just I have explained it. So we have to adopt it in our life. We have to make the aware. We have to launch the schemes of the government. We have to be volunteer of the United Nations. You have to be the active participant in the programs, in the events. In this way, in first step, we can contribute the mission of the United Nations. So I expect in a short period of time, just I have explained it. Now 5 to 10 minutes is needed for the question answers, making the interactions. So in these 5 minutes, I expect the 55 minutes I have contributed. For that, actually, I don't use the PPT programs, so I cannot share it. I explained it, what I have the basic knowledge of it. I contribute directly to the audience, to the scholars, making the interactions. I don't use anything like the laptop, mobile, notes, PPT, etc. So there may be hindrances and obstacles in the transfer of the materials to you. So I cannot carry it, transfer of materials. If you have recorded it, it may be available by the organizer. But if you are in contact with me, my presence is available on the Google search. If you put my name, Dr. Nilesh Narayan Prasad, in Google search, you will get the complete profile. Complete profile means even the email ID, mobile numbers, what is my association, what was the my association, how I am associated with different international platforms, etc. All you will get it. So please come in contact with me. Now the time for the interactions. I need suggestions from your side also. If I will get the positive suggestions, that will be also helpful in the execution of the mission of United Nations. I am in the association with United Nations also. I am acting on it. In the last Science Summit, I was also the part of the panelist in United Nations Science Summit programs, 80 program, UNGA 80 programs. I was the convener also, and I had participated. I am the member of the Food and Agriculture Organization, also FAO, member of this community, agriculture, sustainable agriculture mechanizations. I am also the. So that is the small contributions to the country from my side. I need your support. I am interested for making the collaboration partnerships. If you are interested for the launch and for the execution of the mission of the United Nations. So please, the time of the interactions, make suggestions to me, make contributions. Thank you. Thank you to all.
Such inspiring session.
Hello, are you listening?
Yeah. Thank you for the a very informative session. Um, it was a very useful. I have got couple of questions. Means at the moment the audience we got students, faculty, and also audience from industry, pharma industry. So you talked about SDG 3, but I want to understand that how, as a researcher, as a company, we can also be, thinking these sustainable development goals need to prioritize. So when we look at climate action, that is SDG 13 and SDG 6, that is the clean water. Definitely the companies and also the researchers plays important role there. What will be your suggestion that how, as a researcher, as a company, we should actually make sure that we are trying to follow the sustainable development goals, so that way we can be part of reducing all these
Okay. I understand.
yeah.
I understand. Okay. I have explained, but once again, I am going to say that actually what you do must be noticed. If you do, but that is unnoticed, it has zero importance. So at first, on the behalf of certain organizations, if you are doing the carrying the research work under any university, under any college, research associations, at first we have to register on the portal of the United Nations on the behalf of that organizations. There is the process of registrations. Registration link is available on the portal of United Nations, and you have to put the registration there. And now after the registrations, making registrations, if it is noticed by the United Nations, and you will get the project ID. Project ID, under that project ID, you have to carry a series of programs, awareness programs, carry the webinars, symposium, physical meetings, seminar, online programs, making the collaborations with different organizations, etc. And in this way, you can make the contributions. But if you are not able to make the registration, and now you want to make the contribution to the country, that is a part of your good initiative, you can do it to reduce the harmful gases in the environment. You can take part in the plantations, forestations. You can take part in the plantations. We can plant small plants in your areas, in your college campus, in your nearby areas. You can be the part of this mission. A chain of the NGO are doing the work for plantation, forestation, etc. You can be the part of this. If you are a researcher and the research scholar, you can carry the research. How can you reduce the possibility of the harmful gases in the environment? You can launch the schemes of the government. So many schemes have been launched by the government. One scheme is launched by you successfully. It means you are contributing to the society, you are contributing to the country and for the welfare of the environment, for the complete world. So if you are determined for that, you can carry it at first grassroots level from your village, from your city, from your college, from your university. You can start it. You can start the programs for that. At first, persons, people must be aware. For the execution of the schemes and programs of the government, at first, at first, the section of the people must be aware about that. So at first, you have to take initiative. You must have the association with particular college and university. If possible, make the registration on the portal of the United Nations. In this way, you can contribute to the society. You can launch the SDG programs. You can contribute for the mission of the United Nations. In brief, just I have said that is a long process. It is a continuous process. So many at first pass through the schemes of the government. A chain of the schemes are available. But people living in the remote areas and the villages are not aware about that. Even in the
Not
urban
aware.
areas
Yes.
also, they are not aware about it. So you are requested to launch the awareness programs at first. In first step, launch the awareness programs. Thank you.
Thank you, sir. Thank you for the explanation. And definitely, I think SDG is important, and that's the reason actually we, made it a part of this conference, because we wanted to make sure that the students, the researchers, the companies are aware of what is going on under the SDG. Thank you.
Any more questions? Any suggestions from your side? If you have any suggestions from your side that will contribute
We
for
will
the
share
betterment.
your details, Dr. Nilesh, to the attendees. There will be definitely some people who will be interested. Definitely, Swalife will be also interested, so we will contact you. But there will be people from the the attendee and also from the evaluators who are working. So maybe they can be part of some collaboration. So I will share those contact to those people as well.
Okay.
Yeah, over
Thank
to
you.
you. Over to you, Ruchira. Thank you, sir.
Yes, good afternoon, everyone. So now we will begin with our next session. That will be the oral presentation session. So before inviting our oral presenters, uh, I would like to acknowledge and extend the panel of evaluators who will be judging towards today's presentation. The feedback and scientific expertise will inspire our presenters and ensure a fair and comprehensive evaluation process. Our respected evaluators are Dr. Praveen Berde sir, founder of Swalife Biotech, Dr. Santosha, Assistant Professor at Department of Radiation Oncology, Sri Devraj URS College. Next is Dr. V. Anita Kumari, Professor at Department of Pharmaceutical Chemistry, Sir C.R. Reddy College of Pharmaceutical Science. Let us extend a warm welcome to our respected evaluators. We now begin our oral presentation session. Please ensure that each presenter will have approximately 12 minutes to present their research, followed by evaluation from our esteemed judges. We request all presenters to kindly adhere to allotted times. So, our first presenter is Miss Medisetti Gayatri Devi. I request you to kindly present your work.
Is it visible, ma'am? Hello.
Yes, you are yes, we are visible to you.
Yes, ma'am. Thank you so much for inviting me. Uh, so thank you so much, Swalife International Conference. And my name is M. Gayatri Devi, working as associate professor in Vishnada Institute of Pharmaceutical Sciences in Vishakhapatnam. And today's topic is formulation and evolution of starch-based phosphate moisturizing cream derived from Mangifera indica. So the contents are aim and objective, plan of work, introduction, materials and methods, results and discussion, and summary and conclusion. The aim and objective. The main aim of this project is to formulate and evaluate starch-based phosphate moisturizing cream derived from Mangifera indica, that is from mango kernel powder. And the objectives include extraction of starch, characterization of starch, conversion into starch phosphate, and preparation of cream with different concentrations, and evaluation of final cream. And the next, going to the introduction. Generally, starch is a natural polymer widely used in pharmaceutical formulations, and native starch has some limitations like poor stability during processing. So it is chemically modified into to improve its properties. In this study, mango kernel starch was selected because it is natural, inexpensive, biodegradable, and obtained from waste mango seeds. And coming to introduction about the creams, creams are generally semi-solid preparations applied on the skin for protection and moisturization. So generally, rice starch, many starches are used for the preparation of these creams. Modified starch improves cream properties such as viscosity, stability, smooth texture, and water retention. So that's why we are selecting the mango kernel starch and converted into a starch phosphate was used in the moisturizing cream. Coming to the plan of work, extraction of here in this almost five steps are included. And first step is mango kernel starch was extracted, and then physicochemical properties were evaluated, and then starch was converted into starch phosphate. After that, moisturizing creams formulations were prepared with different concentrations. Finally, the creams were evaluated. So this is the method of extraction of mango kernel starch by using centrifugation method to separate and collect the starch, and the obtained starch was dried and stored for further studies. This is the different steps of centrifugation method for the extraction of mango kernel starch. And this step is conversion of mango kernel starch into mango kernel starch phosphate. The extracted starch was chemically modified into starch phosphate. So this modification improves water binding capacity, swelling ability, stability, and making more suitable for cosmetic preparations. This is the formulations of mango kernel starch phosphate moisturizing cream, different formulations. Here the F1 contained native mango kernel starch, and F2 and F3 contain different concentrations of starch phosphate, and F4 contains rice starch as comparison. Other ingredients like mango seed oil, zinc oxide, cytosterol alcohol, vitamin E, glycerin, preservatives, these are also involved. This is the next step of preparation of cream using the extracted mango kernel starch phosphate. Coming to the evaluation, different parameters like pH, spreadability, viscosity by using a Brookfield viscometer, moisture retention. So this test indicates that better moisturizing performance and a longer hydration of the skin. And coming to the results, many parameters are evaluated like physical appearance, starch yield, and identification tests for presence of starch and angle of repose, determination of amylose and amylopectin, pH, solubility, swelling capacity, water binding capacity. So these are all different types of parameters we have evaluated. So these are the observations like starch yield 72% we got, and iodine test, angle of repose, amylose content, amylopectin, these are all within the range only. And these are the other parameters like swelling capacity, wettability, water binding capacity, dpph for antioxidant activity, and FRAP values also for antioxidant property. This is the FTR spectrum of mango kernel starch. So this confirmed that presence of important functional groups such as hydroxyl glycosidic linkage is showing that the extracted material is a starch. This is the functional groups present in the starch, and this is the mango kernel starch phosphate FTR spectrum. Here thus confirm that successful formation of starch phosphate and indicate improving water binding and thermal stability. These are the functional groups and peaks are present in the FTR starch spectrum. And next one is SIM analysis. This showed that starch granules were irregular, oval, polygonal shape, and two slightly rough surface. So these are like confirmed as a starch granules. So this suitable as a thickening and binding agent. This is the X-ray diffraction analysis showed that mango kernel starch has a low crystallinity with the B type and C type starch characteristics. This property is useful for cream and gel formulations. This is the evaluation parameters of moisturizing cream like physical appearance, pH, spreadability, viscosity, moisture retention, stability. And coming to the summary, mango kernel starch was successfully extracted and modified into starch phosphate and used to prepare a moisturizing cream. The cream showed that good physicochemical properties and promising moisturizing performance. And finally, I concluded that the study demonstrated that mango kernel starch phosphate can be successfully used as a natural ingredient in a moisturizing cream. It provides good stability, spreadability, and hydration while offering an eco-friendly alternative to synthetic excipient. That's it, ma'am. These are the references I followed. Thank you, sir.
Okay, thank you. Now we will go with some questions to you. I request
Sure.
evaluator to have some questions to you.
Sir.
Hello, Dr. Santosha. Any questions from your side?
Hello, good afternoon. Can I see the first slide, sir?
Yes.
Oh, aims and objectives. No questions, sir. Good presentation. Thank you.
Thank you,
Thank you.
ma'am.
Thank
It
you.
was, yeah, it was nice presentation. Just
Thank you so
from
much.
company point of view,
Yes, sir.
you extracted the starch from mango kernels. So I
Yes, sir.
want to just understand what was the costing involved, and if we want to scale up, what will be the costing involved, and how can it can be scaled up?
Because actually it is eco-friendly, sir. Completely wastage product. Kernels are definitely we are not used, so that it is a wastage product. Nearly 100 grams of kernels we will get starch yield. Almost 70% of starch we will get, sir. So it is economical, sir. Actually.
Okay, and the purpose of this starch is like a additive, right? It is not for
Ah,
the main ingredient.
yes, sir. Actually, it is a main ingredient only, sir. It is having an antioxidant property also. So we
Okay, but
proved
that is
also.
not evaluated, right? Or it is evaluated.
We evaluated, sir. We
Okay,
are not included in this PPT, sir. For
okay.
time management, we are not.
Thank you, thank you. It was nice presentation. Good
Thank you
work.
so much, sir.
Thank you.
Thank you so much, sir. Thank you. Thanks.
Thank you, Mehdi Shakti Devi. So now we will move further with our next presenter, Saumik Chatreji. Hello, Saumik. Are you there?
Hello. Yeah, I'm there. I'm there.
Can you share your presentation?
I'm not being able to upload the presentation. Where do I upload the presentation over here?
Dr. Somek, you can directly share the screen as well. It will be easy.
So the option of screen sharing is not coming anywhere. That is the whole problem.
No, if you see, it will show as present or invite. In that, there is an option. Did you find it, or I okay.
To find the option to share the screen anywhere.
Okay, let me show you. I will share my screen. Can you see my screen now? So here is the option, and when you click on that present, there is an option here to share the screen.
Sir, unfortunately, this option is not being getting shown in my iOS app. That is the problem, sir.
Okay, so you join from the mobile, you're saying.
Yes, sir.
Okay, 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 that's fine. Ruchira, can you have you shared your PPT to us?
Yes, sir. Yes, sir. Yes, sir. I've shared my
Okay,
PPT, sir.
Ruchira, can you
Uh, yes, sir. I have done that. It's loading.
okay,
Within
yeah,
5 minutes, we will start.
okay, okay.
I- i- In the meantime, sir, presenter can share the screen.
Sorry. Hello, Dr. Somek. You said something?
So actually file is more than 50 MB, so not getting upload over here.
Ruchira, you can share your screen. Open the PPT on your screen and share the screen. I don't think now he's there. He is log off. So you can keep the PPTs ready. Uh, on your screen, you can share your screen and we can, go through that.
Sir.
Okay. We'll wait for a
Okay.
couple of minutes and if you join, then we will continue. Otherwise, you can take the next speaker.
Okay, sir.
Yeah, he's there now. So you can add him to the stage and you can share the PPTs.
Okay, sir. तुला तिथे दिसतंय का बघ. You see now, sir?
Yes, just go to presentation mode.
Yes, sir.
Yeah. Add him to the stage so he can start.
Yes, sir. He is added now.
Yes, sir, he is added now.
So, please start your
Please start your
presentation.
presentation.
Yeah. Good afternoon, respected chairperson and judges, distinguished professors, senior consultants, and my dear friends. Today, I'm not going to tell you about how artificial intelligence and some innovative studies will replace doctors. But today, I will tell you something which is very uncomfortable, some truth which is very, very uncomfortable. Next slide. So imagine that it's a story of 1 a.m. midnight, not a conference hall, not previous slide, please. Not a conference hall, not a PowerPoint slide, just 1 a.m. midnight. When the city is asleep, when the roads are silent, and when the phone is not ringing normally, it is ringing for emergencies. And the mother is called at the middle of a night saying that her daughter has undergone massive blood loss after delivery of a baby. The monitors are screening, the family is standing outside just praying for blood, and no amount of money can manufacture a single drop of blood. Not gold, not power, not influence, not fame. Even the richest person in the world and the poorest of poor are beggars outside the blood bank. Next slide. And the most dangerous thing about blood that it is not when the blood becomes unavailable, but it is when the blood is given to a wrong person. One wrong decision, one mislabeled sample, and 1 minute of human error can write the last chapter of someone's life. People often think that blood bank or blood transfusion is all about giving blood. No, it is not about giving blood. It's about making the right decision before giving blood. Because every unit of blood can carry two possibilities, whether it can save life or it can end one. Next slide, please. So today, I want to tell that hospitals today, they don't struggle because they lack data. They struggle because humans have more information that they can process. Thousands of reports, multiple tests, compatibility rules, guidelines, and seconds are ticking away in a clinical judgmental error. And that is when exactly we understand that blood can save life, but wrong decision can cost them. And this is where a new teammate enters. It is called as artificial intelligence. It is not to replace doctors, but it is to strengthen their decisions. Next slide, please. Because I believe the future, because the critical decision is to never just say yes or no. It is about to decide whether transfusion is required, which component of blood to be transfused, what is the exact quantity, when is the right time, and will the benefit outweigh the risk? Because transfusion medicine, the future of transfusion medicine is not about replacing human intelligence. It's about making sure whether human lives are never lost because human intelligence might run out of time. Ladies and gentlemen and honorable judges, I invite you to explore how we have discovered that artificial intelligence can transform one of the most critical decisions, the decision of blood. Next slide. Now, why are we so much focused about blood? Because blood is unique. We cannot manufacture blood in factories. We cannot print blood on a 3D printer. We cannot synthesize blood in laboratories. So every single unit comes from human being. That means every unnecessary transfusion wastes a precious gift, right? One decision, two possibilities. So this is what the aim and objective of our studies was, to evaluate whether AI-assisted clinical decision support system could improve transfusion appropriateness, could enhance the decision whether transfusion is required or not, and to reduce the avoidable transfusion-related adverse effect. In a nutshell, AI as a friend to support clinical decision making, right? So we took a sample of 20 patients. These 20 patients voluntarily agreed to participate in the study, and we have run a automated AI assisted clinical decision support making. We have taken help of 3 AI platforms to run the decisions, and the in the 20 patients initially the clinical parameters, laboratory findings, blood parameters, and indications of transfusion was studied by a human doctor as well as a AI assisted model. And after these 20 patients data was collected, we first made a decision based on what the human doctor is suggesting, and we then asked the AI to give recommendations and then compared alongside conventional clinical decision making. So rather than replacing physicians, next slide, rather than replacing physicians, the artificial intelligence model acted as a second opinion. Now, what did we learn from this? That AI will not replace any clinical judgment. It made every decision more informed, right? It rapidly integrated multiple patient-specific platforms and generated structural recommendations. Most importantly, as you can see, the recommendations showed good agreement with the conventional clinical practice. Respected judges and all the people who are attending it, we often see that whenever you write something on ChatGPT or Copilot or Perplexity or any other platform you see, it says that AI can make mistakes. But in this 20 patient sample study, the model was written in such a manner that there is no scope that the even if a decision is made wrong by the artificial intelligence, we take it into consideration because it is always giving us a second opinion, a second suggestion. And this time, the second suggestion is not your friend, colleague, or doctor. The second suggestion is artificial intelligence because every decision, every minute counts whenever it comes to clinical judgment. So AI identified situations where immediate transfusion could safely be deferred, allowing close monitoring. This contributed in better transfusion planning, more efficient blood utilization, and fewer avoidable transfusions. Next slide. Now, what did we understand through all this study and what is the interpretation? Now, what did these findings actually mean? They mean that medicine is slowly evolving from generalized treatment protocols. I'm using the word evolving over here. I'm not saying it is getting replaced or it is getting conventionally deferred. No, it is evolving from the generalized conventional practice towards more precision medicine. That same transformation is happening in transfusion medicine as happening everywhere. It is helping us in early recognition of patient observation. It is helping in improved transfusion planning and most importantly, reduction of unnecessary transfusion because I tell you one single line which I have quoted before that blood is very unique. You cannot manufacture in laboratories. You cannot print it on 3D printers. No matter how advanced we get, it comes from human being. Every single blood comes from human being. So evidence-based patient management is improved through this transforming clinical decision making through AI. Next slide. Next. So AI prevents adverse transfusion reactions. It identifies which patients are at risk of adverse transfusion reaction. It predicts whether there is allergic, febrile, hemolytic reactions and transfusion associated injury because every blood component carries risk, right? Every blood component carries transfusion reactions, volume overload, alloimmunization, infection. So the safest transfusion is not the fastest transfusion or how quickly you decided whether transfusion should be done or not, but it is the right transfusion. It is whether transfusion was at all required and which component of transfusion was required in this case. Next slide. So please remember that artificial intelligence is efficient for blood component utilization as well because it optimizes the inventory management, right? There is we have whenever there are human to write different components of blood components available in the inventory or in the storage, it can make mistake. But once you have data through AI or integrated models through AI, automatically whenever you collect blood, every component separation and whatever component is there is right visible in front of you, right? Every person, be it in ICU, be it in general ward, be it in intensive care unit, be it in oncology unit, be it in radiotherapy unit, radio diagnosis, any person, pathology, microbiology, any and every person from any part of the hospital can see which blood component, how much blood component, how many liters of blood are available, how many blood packs are available. So everything is automated and every inventory is automated because AI is as intelligent as it data receives, garbage in, garbage out. So therefore, it supports clinical expertise in transfusion medicine. My dear friends and my senior consultants and judges, please, what I'm trying to focus over here is in transfusion medicine, blood does not make mistakes. Decisions do. So every unit of blood carries one question: Am I about to save blood or am I about to end one? Next slide, please. Because we have to understand that transfusion is not only a procedure, it is a judgment. It is a judgment. Like every study has some limitations, we too had certain limitations. Our limitation is that it included 20 patients because it is the first ever study done from West Bengal and Eastern India. I want to highlight this. This is the first ever study done from this part of West Bengal, probably this part of the eastern India. We included 20 patients because any model, integrating any model on AI is a cost effective thing, and we had to bear the cost all on ourselves. So the 20, because it is not, we did not get any fund from any agency. So since it was self funded, so only 20 patients could be included in a single center based study. But this limitation is something which is a blessing, because in 20 patients which we counted, in all 20 patients, AI showed 100% efficacy. 100% efficacy. When the study was done, it is a single center study, but now we have done in multiple center studies, and we have integrated with the hospital EMR records as well. In future directions, we can include in any which hospital, like if the receptionist is making the if receptionist is want to know how many units of blood are available, she can see. The company, the hospital owner can see, the hospital superintendent can see, the intern, house staff, everybody, PGTs, postgraduate trainees, everybody can see through the records available. It is integrated to the hospital's own record. And real-time predictive transfusion support is something which we are looking forward to it. Can we move on to the next slide? Because if one wrong decision can cost a patient's life, imagine the value of a system that will help us to make the right one. Medicine has always relied on human intelligence, and today human intelligence has a friend, that is called artificial intelligence. From complex data to
Soumik,
precision decisions, right? Yes, ma'am.
you only have 2 minutes more to present
Yeah,
that.
yeah, yeah. I'm just about to finish. So, patient
Yes.
assessment and clinical history, vital signs, and all the clinical data are comprehensively integrated in this AI model. And just by it is how simple. I will show you how simple it is. You can just enter the patient's laboratory details, the patient's blood group, patient's current hemoglobin level, and indication of a transfusion. The AI model will ask you a couple of questions to help you to understand whether decision is done or whether correctly transfusion is done or not. And as and when the questions are answered, you will come to one single query, that whether transfusion is required or it can be deferred. Next slide, please. Artificial intelligence, I'll tell you, my dear audience, artificial intelligence will not replace clinician. It will protect the patient from limits of time, complexity, and human oversight. Right? The future of transfusion medicine is not smarter machines. It is about safer decisions. Because the most expensive mistake in transfusion medicine is not measured in money. It is measured in the heartbeats that never return. Next slide. That is what I'm trying to say, that artificial intelligence will not replace clinician. It will protect the patient from limits of time, the limits of complexity, and human oversight. One unit can restore a heartbeat, and one wrong unit can silence it forever. In medicine, seconds matter, but in transfusion medicine, decisions matter even more. Next slide. Because we know that blood has the power to save a life, and decisions have the power to decide whose life will get saved. So artificial intelligence combined with clinical expertise will improve and enhance the outcomes of precision in transfusion medicine. That is what we try to say, that from algorithms to lifelines, the precision, the importance of artificial intelligence integration in transfusion medicine. Next slide, please. And I would like to conclude by saying one single line, that a bag of blood is only 350 milliliters, but the decision behind it can carry the weight of an entire lifetime. Thank you so much for listening. That was my time. Thank you so much.
Yes, thank you, Soumik. We will now go with some questions.
Good afternoon. Yeah, it's a good presentation, a clinically relevant topic, as transfusion errors is a preventable cause for the mortality and morbidity. I just want to know what is the methodology for this study?
Sir, in 20 patients, we have first incorporated the data which we had, that what was the indication for transfusion prescribed by a physician or clinician. And then the medical officer on charge of the blood transfusion department was made to assess whether transfusion was required or not. The same set of data was then integrated in integrated AI platforms, and then we have primarily we have taken help of Perplexity and Claude. And Perplexity and Claude, we have given our own learnings and codings, and then we have set up certain questions. That certain questions will be filled. What is the hemoglobin level? What are the indications whether the patient had previous transfusion, the last indication for the last time he had a surgery, alumination, everything. And once the entire data is fit in, it will say whether transfusion is required or not. And we have correlated these two data: the human, the medical officer in charge of the blood transfusion department, as well as the AI, we call it the AIO, AI officer in charge of this model. And then we have compare decisions. And sir, you won't believe, in all the 20 decisions we have seen that when AI help was taken, it successfully reduced the errors as well as prevented transfusion which was not required. 1 minute of your time, sir, I'll share one story because it's first study from West Bengal. In one patient, the alloimmunization part was overlooked by the medical officer in charge because he was a young intern. So he overlooked that thing and he was about to write that transfusion to be given. Immediately, the AI said that alloimmunization is the history of RH incompatibility is there. So we could have saved the patient's life. Otherwise, the patient would have gone into ICU and maybe landed in the post-mortem unit. So we have saved the patient's life just by this one single error. The intern was not trained enough to take the decision. But in resource constraints training, sir, my major focus was in resource constraints trainings. This could be used at a massive scale.
Yeah, that is fine. Like while issuing the blood, in the blood bank they will do all the all the testing, cross matching, everything. Once it is compatible, then only we will transfuse the blood to the patient. Okay. And even if it is there, life-threatening complications are very rare
Sir,
until unless
sir, not
sterile,
being able to, sir,
until
not being
unless.
able to hear you, sir, not being able to hear you. Can you please?
Can you hear me now?
Yeah, yeah, yes, sir, yes, sir, yes, sir.
Yeah, so I was telling, like after cross matching only we will issue the blood to the patient for the transfusion. Sometimes there will be some reaction, and until unless it is a different blood groups or transfused, the mortality is very limited. So immediately, proper care will be taken. Like AI definitely will help us to prevent all these things. Good presentation.
Thank you so much. Thank you.
Thank you, Dr. Somek. It was a nice presentation. There are a couple of questions from my side. So, when you say you collected samples from patient, do you mean you collected blood?
Yes, sir. Yes, sir. Yes, sir. I mean,
So this,
we don't collect blood as a whole for to analysis. What happens is that we have certain blood storage units, which I mean, blood is collected from some blood donation drive, and then it is stored for transfusion, right? And we collect patient's sample, which is given for routine investigations like CBC or other parameters. We don't collect separately for this analysis.
so this protocol was sanctioned by the committee, right?
Yeah, yeah.
So, under what type of clinical trial this comes?
Sir, this is not a clinical trial. We have not experimented anything on the patient. We have
So,
not
observational.
done any observational studies, observation, because we have just tried to incorporate that whether decision was done or not. At the end of the day, since it was a small study, at the end of the day, we went with the decision the doctor has taken, right? But every time an error has taken in all the 20 patients, there was some error. So we consulted a hematologist. We consulted a transfusion medicine specialist and the pathologist as well. And then because I come from the pathology department, I am a postgraduate trainee in the department of pathology myself and transfusion medicine myself. So now we analyzed whether it is actually required or not, or whether AI is doing the correct thing or not. So at the end of the day, in this study we have done a human study only. But when the human judgment is taken into consideration for transfusion, because at the end of the day we are responsible for the patient care. But it is we don't have not done any intervention based on what AI has just given us. But the case we highlighted that it was an alloimmunization, the intern overlooked it, but when we checked it, we see there is a history of alloimmunization, so we have to taking care of our RH incompatibility, and we stopped or deferred transfusion there. So that is how we have taken into this consideration, and we have taken consent from all the patients.
Okay, and what type of model you used? The AI
Sir,
models.
so we have used machine-based learning for ChatGPT 5.0, Perplexity, and Claude.
Okay.
All,
Okay.
all of
Thank
them,
you.
all of them in their paid subscriptions, and no free models. All of them in the paid subscription and fully integrated. Because we have one person in the team who's masters in artificial intelligence and data science, who has drafted the final code for it.
Okay. Okay. Yeah. Thank you. Thank you. It was nice
Thank you,
presentation.
sir. Thank you,
Thank
sir.
you.
Thank you. Thank you, sir. Thank you.
Thank you, Samir. Now we will move further with our next presenter. So our next presenter is Dr. Nagabhati. I request Dr. Nagabhati to start their presentation. Am I audible to Nagabhati? Please turn on your mic.
I am audible, madam. Hello.
Yes.
Hello. I am audible, madam.
Yes, yes. You are audible now.
Good afternoon, everyone. This is Nagabharati. I am working as associate professor in Vigyan Institute of Pharmaceutical Technology, sir. I am from Andhra Pradesh. So I am very thankful to these organizers and the panel members for this opportunity. And I am starting my presentation, sir. So my topic is granisetron. We have repurposed this granisetron. It is an anti-emetic drug, and nowadays we have lot of things are going in drug discovery during regarding this repurposing. So we have tried this granisetron, which is serotonin inhibitor, for repurposing it for the Alzheimer's disorder, where we see this Alzheimer's disorder has been increasing drastically from next slide, madam. Next slide. Next slide. Please change the slide, madam. Here we can see the burden of the Alzheimer's disorder, sir. So here global burden already 50 plus million people have been suffering, and it is estimated that by 2050 globally the rate has will be tripled, and cost for this therapy also is getting increased. So in this regard, what we have thought is the main aim and objective for this project is, if you try to repurpose a drug which is already established, so we can reduce the cost of drug discovery, and we can get the drug for a lesser price in short duration of time. That was the main objective for conducting this study. Next one. And one more thing is, whatever the drugs are there, they are only clearing the symptoms. They are not completely treating the disorder. So this is our main motto regarding the conduction of the studies. Next slide.
You can change your slide from the screen. Can you?
No, ma'am, I am unable to do it.
Okay, let it be. I'll do. Okay, move further.
Yes, so as I have mentioned, drug repurposing is one of the most cost effective, and granisetron, I have mentioned, it is a 5-HT3 antagonist, and this is our main intention for using this drug, and it is also used for vomiting. So our main intention is, as Alzheimer's is a central nervous system disorder, so this drug can be used, and for crossing the blood-brain barrier also it will be useful. So this is our hypothesis. Granisetron may inhibit the anticholinesterase and butyrylcholinesterase. These are the two enzymes which are present in the brain. And when, if you see the pathophysiology of Alzheimer's disorder, there is an imbalance between acetylcholine. So more destruction of acetylcholine. So Alzheimer's has been occurring. So if you prevent this enzyme, we can increase the concentration of acetylcholine in the brain, and we can improve the symptoms of dementia, that is memory loss, and other symptoms. Next slide, madam. So we have used three approaches here, sir. Already everyone are into the in silico models. In vitro, we have done an assay, and ex vivo also we have performed by using chikungunya. Actually, why we have used chikungunya is in in RRR policy, that is recycle reduction in the utilization of animals. So chikungunya was a good model we thought, and we used this. And for in vitro validation, we have done Elman's method, which is for cholinesterase activity, where we have identified the IC50 values, and in silico method for our molecular interactions we have done, and we have used a software called as Pyrex. Next one. So this is the computational results. Actually, tacrine is taken as a standard drug for this study, and granisetron, this is our compound which we are claiming that it might have the activity. So when we have taken this protein molecule and we have started the docking, we have seen this binding energies. Here, if you see the binding energies, the more is the negative value, the stronger is the binding. So as standard drug, tacrine is showing minus 6.75, and our granisetron is showing minus 7.92. So it indicates that it has more affinity towards these enzymes, and we also concluded that these are the residues with which it is interacting more, and it is having a good binding affinity for that, so that it can be used in the late stage Alzheimer's, where it can demonstrate this inhibitory activity, so that as I have mentioned, acetylcholine can be maintained. Next slide, madam. As I have mentioned, we have performed Elman's method for identifying IC50 values. So this is with our granisetron, and this is with our tacrine, and these are the IC50 values. Actually, IC50 means minimum inhibitory concentration. So here we have seen the inhibition of acetylcholine and butyrylcholine esterase by using a UV method. Its method name is Elman's method, and here we measure the absorbance by using UV spectrophotometer, and IC50, it is 3 to 4 micromolar in the case of granisetron, whereas in the case of tacrine it is 8.5 micromolar. So it is indicating that granisetron is having a good potency in inhibiting this enzymes. Next one. And we also performed ex vivo also, sir. So there we have used the chikungunya preparation. Actually, I have not represented all those diagrams. And for Elman's method, we have done enzyme isolation. We have isolated acetylcholinesterase and butyrylcholinesterase from the blood cell, human blood. And this entire protocol was approved by our institutional ethical committee. And this is with your chikungunya, sir. And what we have observed is, we have observed a dose dependent um, enhancement of the decrease in the contractility. So here we have noticed that 16 mg per ml is the optimal dose, and after increasing the dose after certain dose, we have also seen the sensitization of the receptors, and no, there was no further increase. So there is a decline in the contractions. So in the graph it is been depicted, and whenever we compare our granisetron with physostigmine, granisetron has demonstrated a higher potency, and it has inhibited the acetylcholine esterase more than the physostigmine. So in this way, what we are claiming is, with our study, we are saying that granisetron can be used as a repurposed drug for the utilization in the Alzheimer's disorder. Next slide, madam. But next slide. So these are our key findings, and what we have observed in this study. So computational productions are aligned with our in vitro and in vivo experimentational evidence, and because of this inhibition of butyrylcholinesterase, we have a particular advantage of this using of this granisetron in the late stage of Alzheimer's management. And we also founded that lower IC50 values means more potency of the drug, so low doses of granisetron can be used than the tacrine. So, and again, we have inhibited both the enzymes, so which are important for the management of your Alzheimer's disorder. Next slide. So, as all we know, we have a few limitations in this study also, and as it is 5HT3 antagonist. It can cross the blood-brain barrier, but granisetron is partially having this BBB hindrance, and so we have to see into this parameter, and we can also go into further studies validating it in animal models as because of limited resources in the college we have done only with the chikungunya. We can go with the nanoparticle like preparation or liposomal delivery systems for the enhanced movement of this drug into the brain, and we can also use it in the Alzheimer's effectively. Next slide, madam.
Naga, you only have 1 minute more to present, okay?
My slides are completed, I think, madam. Next,
Okay.
actually, coming on to my conclusion. So, with this computational as well as in vitro and in vivo evaluations, what we have identified is we have identified that granisetron is having a dual effect on acetylcholinesterase and butyrylcholinesterase. So, because of this dual inhibition, it is having greater potency, and it is also addressing your butyrylcholinesterase inhibition also. So, it can be repurposed, and as it is already used as a drug, so repurposing is an easy process, and we can decrease the timeline of the drug entry into the market, sir. So this is what I want to say. Next are references. Thank you, madam. I have finished my presentation, and I am open for the
Okay,
questions.
thank you. Thank you, Naga. Now we will go with some questions. I
Yes,
request evaluators to have some questions.
yes, ma'am. Yes.
Hello, good afternoon.
Good afternoon, sir.
Yeah, good presentation, good study as well. What is the percentage of improvement in cognition in granisetron group?
Actually, sir, this is not a clinical study, sir. We have not extended this into the clinical trials. We have done with only in vitro and in vivo. We have seen whether the acetylcholinesterase and butyrylcholinesterase are getting inhibited or not.
Okay.
We haven't went into the clinical trial perspective.
Okay.
That is the future direction.
Okay. Good presentation. Only complaint is like slides were not visible as like they're like very small. I'm not able to read only.
Okay, okay, sir.
It's otherwise it was a good presentation. Okay. All the best.
Okay, thank you, sir.
Thank you, madam. It was nice presentation. Just just one question. Is there any evidences available where your drug has been already utilized in Alzheimer disease?
Sir, there is a paper reporting regarding ondansetron, sir, not granisetron.
Okay. So,
Ondansetron was already reported. They are going for clinical trials. They are in the phase 2 and phase 3, sir. But granisetron, nowhere it was. So we have tried, sir.
okay. So uh, the hypothesis was built on that.
Yes, sir. Yes, because both are serotonin antagonists. So ondansetron is being used. So we want to see whether this is also working or not.
Okay. And are they used in like for any effect which are happening in Alzheimer, like nausea, vomiting? Are they used at that time?
Definitely, they can be used, sir, because their current indication is that only. Current indication is they are used in the cancer chemotherapy for vomiting and related nausea. So, as it is already means already marketed for that purpose, but we repurposed it for Alzheimer's.
Okay. Okay. Thank you.
Thank you, sir.
Thank.
Thank you, Ananda.
Thank you so much. Now we will move further with our next presenter. So our next presenter is Jitendra, and hello, am I audible?
Yeah. Yeah. You're audible.
Okay, so now our next presenter will be Parbali Mitra. Is Pamadi Mitra available here?
Yep. I'll just give me a minute. I'll share my screen.
Okay.
Is it visible? Is
No.
my screen visible, ma'am? Not yet, right?
No, not yet.
Okay, just just a minute. I think I'm not very familiar with this one. I hope this is visible now. Is it visible now?
No, ma'am. We are not. One second.
Okay, just a
We will
minute,
add you on
because
stage.
I think it's visible because it's showing that it is.
Yes. 1 second, ma'am. We will add you on stage. Your
I
presentation will
think
be
that would be
added on
better.
stage.
Yes, please do that. I'm not also very familiar.
You can just slide it. Yes, ma'am. Now you can start.
Thank you so much. Thank you so much. I hope everyone in the audience is able to view my screen. So I am Purvali, and I don't know if this fits the conference theme exactly. It might be a little divergent, but yep.
Yeah, just go in the presentation mode, ma'am.
Yeah. So, but I think it adds to the diversity of the theme. So, the research idea that I'm presenting is titled "I'm Not What You Think I Am," and it explores the limbo between human and the post-human. And I'll be sort of focusing on the cinematic text of Terminator: Dark Fate, which is a movie. I don't know if people have seen it. It was released in 2019. So, I'll quickly run you through the abstract. Basically, what I'm trying to say is that with upgraded biotechnology, the corporeal space, the human space, has been liberated. So the body can now become redundant almost. And we are all living in a world which is a world of flux and overlap. There is a lot of pluralism and hybridity and multiplicity. And this cinematic text that I'll be exploring, that I'll be looking at, talks about how humans in this world have become non-agents, which means basically it's a world of dysfunctional humans, but rather robots are those who have a surprising amount of agency. So I'll just quickly run to the next slide. So the purpose basically of this is to look at, as I said, how identity categories, subjectivities have become more complicated and complex, because we are exploring new hybrid forms of consciousness, alternate realities, parallel universes. And in all of these, what I kept on saying, that my focus is looking at how in this particular cinematic text, humans seem to have become almost non-agents. They have no power, they have less willpower, and less choices available, whereas robots seem to become more and more powerful. And this slippage of subjectivities basically point out to this one very good question, that whether is there any essentialized human nature, whether we do have to believe that there is an essentialized human nature which has to be saved from simulation and augmentation, and whether it's all a facade, and whether there is a splitting and alterity at the core of humanness. So here, every organism, cyber, non-cyber, seems to be fragmented, and this is of course a world of flux and fluidity. So this is the poster of this movie, this Terminator, and I don't know if people have seen it yet. I'll just give you quickly run you through the story. So two white upper class women, Sarah Connor, which is the central character of this franchise, if people have seen other Terminators. She and Grace, augmented human soldier, cybernetic soldier, they try to protect a Mexican migrant worker and from deportation, and also from a cybernetic patriarchy. And in this mission, T800, a robot aids them. And here again, as I was saying, we are looking at how humans are non-agents. This text actually exposes humans as powerless and fragile, and how they cannot fight a world of the rogue AI without the aid of robots. So robots here are Grace and T800 are the robots who are helping the humans fight the cybernetic patriarchy. And of course, this question again, that whether there is an essential human nature, and this is exactly a cautionary tale that one needs to look at, as how an accelerated post-humanism, we are moving from human to post-human, so it can cause extreme and problematic ruptures in the core of you, the whole idea of humanness, humanization, and all the all the subjectivities are spilling over. There is an overlap, there are hybrid identities, and one must acknowledge that there are non-human subjects. So basically, Grace is, as I said, a white cyborg. She was a human soldier. She cybernetically augmented herself, and now has an high amount of agency. And they together, along with Sarah Connor, who is a complete human, of course, they are trying to kind of replicate a functional lesbianism and trying to act as parents to Dani, who is this multi-positional subject, who is a hybrid subject. She's racially hybrid, and she's a Mexican, so that's why there is also an immigration subtext that's at play in this cinematic text. But that's a different theme, of course. I'm not looking at that. So this racial hybridity meets headlong with cyber hybridity, and there is a borderless society which is kind of promulgated or proposed. And this is very interesting, of course. The character of T800, which is played by Arnold Schwarzenegger, which is called Carl here. And he was actually a self-aware Terminator who was sent to kill John Connor. And then what becomes to his character is he lives as a human later on. He almost adopts a human family and a Mexican woman whom he falls in love with, surprisingly, and as a son. And he slowly is trying to evolve as a human. So I have included a quote which says that after the mission was finished, I had no purpose, and for 20 years I kept on learning how to become a human. So of course, as I said, that identities are constructed in tandem to power, and again identities are also constructed in tandem to technology. I was talking about upgraded biotechnology, and with this overabundance of AI narratives, of this hodgepodge of real and imagined identities, of hybrid consciousness, it has become all part of these fictional universes, this science fictional, speculative, fantastical universes. And the semantic terms, I was talking about identity. So this persistent obsession over identities is also one of the focus of my research idea. Here we see, if you have watched the movie, you'll see that such semantic terms like nobody and somebody are used like almost crazy, like like a refrain. The term nobody is used, overused. I'm nobody, Dani keeps on saying. And my research idea is titled I'm Not What You Think I Am, which is T800's famous proclamation, the Terminators. And then, of course, at one point, Sarah looks at Grace and says, you must be somebody, that you're protecting her. So this obsession over semantic terms as nobody and somebody and, is overused rather. And then, of course, I'm almost drawing to the fag end of my presentation. So again, there is an image and there is a quote that I've never seen anyone like you. And T800 is addressed to by the human characters. They're saying you are almost human. So, of course, this is a cinematic universe. Again, it's fictional. It imagines humans to be dysfunctional and the post-human, entities to be agents. And again, the question whether there is an essential human nature, whether it needs any protection from simulation, or that everything has become overlapped and there is a hybrid, identities, whether they are hybrids only. This is something very interesting, and this is an observation that has been developed in this research idea. And again, I'm depending on the cinematic text, Terminator Dark Fate, released in 2019. If people have seen, they'll be able to understand what I'm trying to say. And which is another interesting thing is that the character of Dani, who is a Mexican immigrant, who is a person without papers, she's an illegal immigrant, and it's interesting that she becomes just a project. Because she doesn't have any agency, though she is kind of pitted again for humanity against a cybernetic world to become the savior of humanity. She isn't actually saving anything. She's just a project of these two white women. And without Grace, the cybernetically augmented human soldier who has now become a cyborg, and T800, nothing could be achieved. This movie tells us that they are the ones, the robots, who are mimicking human agency. They have extreme willpower, supreme choice, and are ethical. because I talked about T800 and his aspiration to humanization. So I'll just end with this thing where I have included in this. I know my slides talk a lot and sort of present are less graphic, but I'll just read you through. So basically, the idea is that Dani Ramos, I was talking about her as an undocumented Mexican, her desire is for naturalization and citizenship, while T800s is for humanization. And I'll just move to the end. Uh,
Uh,
and as I conclude, I would again repeat that I'll repeat with Stephen Herbrechter's comment on the nature of science fiction, where he says that science fiction actually helps dissolution of dismantling of ontological foundations. The distinction between organic, organic, masculine, feminine, human, non-human seems to break down. And as of course I talked about how body can become redundant at one point. Already science fictions like Terminator is imagining that. And there is an hybridity that I was talking about, the slippage of identities, and this blur the essentialized sort of demarcations of the binaries of human, post-human, racial, majority, minority, hetero, homo, center, periphery, which have always kept, traditionally this binary has kept cultural narratives politically conservative, racially privileged, and of course sexually very prescriptive. And this cinematic text shows us that robotic subjects have an amazing amount of agency. I think I'm ending here because my presentation is done.
yes, yes.
Thank
Thank
you
you,
so much.
Parbali. Yes. Now we will go with some questions. I request our evaluators, Dr. Santosha and Dr. Anita ma'am, to proceed further with questions.
Right. Good presentation, madam.
Presentation ready. This presentation, timely presentation, timing also very good. Time management is also very good in the session. Very well managed session.
Thank you, madam. It was nice presentation. I got a question for you. Was the presentation trying to highlight the evolution of robots?
Exactly not, because if you have seen the other Terminators, the robot was always there. The robots have always been there as kind of antagonists. But the interesting evolution that you pointed out is them as being on the side of the humans, trying to fight a bigger kind of challenge. So probably the robots are now evolving into ethical beings. Thank you so much for the question.
Last conference we had in April, we actually had a group from UK. They have developed a robot which has been trained on large language model. So any scientific articles which are being published, they can actually train that robot for that topic, and that robot is actually going to answer similar to what ChatGPT and all other things does, but it is talking. So what we done, we actually asked the question from here, from India, and the robot was sitting in UK, and it was answering the questions relevant to the topic which it was trained. So why I asked this question was the similar reason, means that you gave the examples from the from the movies, but what we have experienced from the research point of view, from pharma point of view, that with the large language models, with the evolution of the artificial intelligence, and also the previous presentation we have seen about the clinical decision making, uh, I think that is where the robots are going. They are evolving, and definitely they will be contributing as part of the means. In our case, it will be research, but simultaneously with different areas, they are already contributing. That is what we think, and your presentation actually reflect that. So thank you. It was a topic was little bit interdisciplinary, I will say, because it is an annual conference, but still it was connecting with the work which the pharma and the researchers are doing. Thank you.
Thank you so much, sir. Thank you.
Thank you, Purvali. Now we will move further with our next presenter, that is Jitendra and Ramya. I request both of you to start your presentation.
Hello, ma'am.
Yes, yes.
Is my screen visible, ma'am?
Just a second. Yes.
Okay, good afternoon, everyone. I'm Ramya. I'm doing PhD under the guidance of Dr. C. H. Jitendra. So, this is my title Multi-Target Modulation of Insulin Resistance by Perilla fructus, and Insight from Network Pharmacology and Molecular Dogging. So, coming to introduction, metabolic disorders are intricate and it is caused by multiple factors such as insulin resistance, dysregulation in the lipid metabolism, chronic inflammation, and hormonal imbalances. Despite the availability of various therapeutic options, it is often failed to understand the root cause of the disease. So, it leads to suboptimal therapeutic outcomes and also high failure rates in preclinical trials, clinical trials. It leads to drug discovery and development of effective therapies. And nowadays everyone is doing for single target and single compound and single disease related pathways. So with the help of this network pharmacology, we can go for multiple compounds and multiple targets and multiple disease related pathways to increase the therapeutic efficacy. So this is my aim and objectives. Aim was to develop the system pharmacology framework to identify and optimize the therapeutic interventions in the treatment of type 2 diabetes. So these are my three objectives. First objective is to identify the bioactive compounds and their molecular targets associated with this perilla fructus plant. And second objective to identify the targets associated with the disease. And for them, I was doing different steps like protein-protein interaction and gene ontology. At last, I'm going to identify the hub compounds and hub genes. And my third objective was I need to find out the more binding affinity to the good hub compounds and hub targets. So present situation, around 589 million worldwide cases are there, and it will be projected by 643 million in 2030. And in all diabetes, type 2 diabetes is 90%. And in 2021, around 6.7 million deaths are reported worldwide. So actually, this type 2 diabetes is a multifactorial metabolic disorder. It is caused by insulin resistance and pancreatic beta cell dysfunction, and it progress is increasing by the chronic inflammation and oxidative stress. So this perilla fructus plant, it is consist of more amount of bioactive compounds like rosmarinic acid, luteolin, beta sitosterol, and some other compounds. So it have antioxidant and anti-inflammatory properties also, and it have insulin sensitizing property also. It consists, thereby it is increasing the glucose uptake, and it is also increasing the beta cell function. So this is my plan of work. The first step was compound screening. So here I used TCMSP database. By applying this oral bioavailability and drug likeness parameters, I'm going to screen the compounds. And after that, second step, whatever I screen the compounds, I need to find out the targets by using these three databases. And third step was to find out the targets related to disease by using gene cards and immune databases. And after that, after collecting all the targets from disease and drug related, I'm going to find the overlap targets, means common targets. So for that, I'm going to construct network by using cytoscape and protein interaction network analysis then gene ontology and tag analysis. After that, at last step, I'm going molecular docking. So this is my methodology. So this is the result. So in the first 1 to 3 steps, around 128 phytochemicals I was screened, but final these 19 compounds are passed by ADME properties like oral bioavailability and drug likeness. And for that, I collected 204 targets from TCMSP and IMPACT databases. And disease targets also 11,134 therapeutic targets I was obtained from gene cards, and OMIM 12 targets. And after removing the duplicate items, at last I got 11,142 targets. So this is the image of the overlap targets. By using the Bunny tool, I did this overlapping targets. So 176 overlap genes I got, and for that I constructed the network construction by using the Cytoscape. So by using CytoHopper plugin software based on degree centrality, closeness, and betweenness centrality, so these 5 compounds are obtained. So degree centrality means these shapes are known as nodes, and these lines are known as edges, which is connecting by the nodes. So degree centrality means if you take any one node, it has direct connections to other node. So based on degree centrality, I was selected these 5 compounds. Next, for the targets, I constructed protein-protein interaction also. So after constructing the protein-protein interaction by string database, I imported into Cytoscope for identification of the hub targets. So these 4 targets I was selected. So same for these 4 targets has more degree centrality. That means TP53 is AKT1, EGFR, and interleukin 6. If we regulate these targets, type 2 diabetes progression will be reduced. So this is gene ontology and KEGG pathway analysis. In gene ontology, 3 steps are there, like biological process, molecular function, and cellular component. So biological process means whatever I got 176 common targets, it will tell in which biological pathway it was most enriched, and in molecular function in which biochemical pathway it was enriched, and cellular component means particularly these proteins are where it was present inside in the cell and what it is going to do. So here, based on fold enrichment and FDR, response to oxygen containing compound and nitrogen compound, these are mostly our genes are mostly enriched in these pathways. Means, for example, reactive oxygens during hypoglycemia condition, mitochondrial dysfunction is happening, it releasing reactive oxygen species. So it is causing pro-inflammatory cytokine production, and also it is causing the cell death, beta cell death also. Thereby it is showing its role in decreasing of insulin secretion, and also it is causing the insulin resistance. And molecular function also, alpha adrenergic receptor activity. So in beta in pancreas beta cells, these alpha 2 adrenergic receptor is located. So if more activation is happening, it reducing the insulin secretion. If there is no insulin means there is a decreasing amount of glucose uptake we can see. And cellular component also in membrane raft. So these are composed with the phospholipids. Generally, insulin receptor is located in the membrane, transmembrane. So if any cholesterol is deposition is happening, it is disturbing the signaling mechanism. So in this pathway, our genes are mostly enriched. So this is the molecular docking analysis. So for TP53, luteolin has more binding affinity, and AKT1 also luteolin has more binding affinity. Interleukin 6 got beta sitosterol has more binding affinity, and EGF for this protein also luteolin has more binding affinity. So when I'm seeing these compounds, mostly epigenin, luteolin, beta sitosterol has more binding affinity to these targets. And I validated this docking, I did the co-crystal validation also. So this is the result of the molecular docking interactions. So in coming to future scope, I did formulation, nano formulation studies and in vivo studies also. Why? Because here I got beta sitosterol has more binding affinity, but it have low solubility and low bioavailability. So I'm planning to develop nano formulation so it can increases its bioavailability. That's it, sir. Hello, sir. Presentation is completed, sir. Any doubts, sir?
Uh, yes, thank you, Ramya. Now we will go with some questions. I request our evaluators, Dr. Santosha sir and Anita ma'am, to have some questions.
Hello.
Yes, ma'am.
May I ask one question?
Yes, ma'am.
How do the bioactive compounds of perilla fructose interact with the different types of targets you mentioned in the presentation, which are involved in
Actually,
the insulin resistance?
how these targets is showing its action on insulin resistance, ma'am?
Interaction with the target.
Compounds are target role in insulin resistance. I
How
didn't.
may different types of molecules may be there, na?
Yes,
How
ma'am.
they may interact with the targets? Any types of interactions, ma'am, with the targets? What are the general types of interactions?
Actually, I selected these compounds based on interaction score, ma'am. Um, for example, if beta cytosterol and interleukin 6 has more binding affinity, so it can decreases the binding of interleukin 6 to interleukin 6 receptor, so that it can decreases the further downstream signaling mechanism activation. It is preventing the cell death, or it is decreasing the inflammation.
Okay. So one more. Uh, which may be the key proteins associated with insulin signaling?
AKT1
What are the vital proteins generally related to insulin signaling
yes, PPI,
pathways?
gamma, AKT1, and PI3K. These pathways are mostly interlinked, ma'am. GLUT4.
And how they may contribute towards the glucose treatment, diabetes treatment or management?
These targets
How
role.
they may manage or they may contribute towards the improvement of glucose levels, monitoring of glucose level?
Actually, I didn't understand your question, ma'am. You are asking these targets role
So
in.
interactions you mentioned, na, ma'am.
Yes,
Interactions you mentioned. Yeah? So how these interactions may contribute towards the
okay.
improved glucose
AKT1.
monitoring in the type 2 diabetes?
Okay, ma'am.
How
If
these
I'm
interactions contribute towards the management of glucose levels?
Okay. If I'm talking about this AKT1, ma'am, for example, if any luteolin or epigenin is binding to the AKT1, so whenever this AKT1 phosphorylation is happening, it is increasing the phosphorylation of protein, A16 something protein is there, ma'am, so that it is increasing the GLUT4 translocation. If more amount of GLUT4 translocation is there, it is promoting the glucose uptake into skeletal muscle. Thereby, it is reducing the blood glucose concentration.
Okay. One more. How the molecular docking studies of different phytochemical constituents from this plant you mentioned, and how they support the identification of different types of targeted therapeutic drugs in the management of type 2 diabetes.
So here for the target identification also I did cytoscape analysis, cytohubba plugin software. So these four targets I got means it have more interaction with the compounds and disease based on that cytohubba analysis, ma'am.
Okay. Okay. Okay.
Okay, thank you, Ramya. Are there any questions more? So, or should I move further?
Yeah, just one question. So, Ramya, you are doing this master project or your PhD project?
Actually, it was minor objectives, sir. I'm doing PhD project only. So I
Okay.
have so many objectives here. I presented only small bit.
Okay, just the question regarding network pharmacology. Do you think that it is a really useful methodology? Because if you look at your targets, if you look at your leads, you are actually taking them from the which are already there in the databases. And when you are
Yes, sir.
targeting a disease like diabetes, which
I did.
actually, yeah,
Yes, sir. I did network pharmacology. Why means, so previously I was doing blindly for pharmacology studies, like I'm taking plant and I'm doing any activity. But with the help of network pharmacology, we may predict either it may be some good effect is there or there is no effect. So that's why I did one
but
step for my studies, sir.
that you can do using the AI now, isn't it? The AI is a predictive and the generative model. It will be more useful because if you look at what you have presented, as you know, you already had a question from ma'am that you know how the targets came and what are the multi leads. You can
Yes,
see your docking studies. In the docking studies, the binding affinity is very low.
yes, sir.
Yeah, it's very low. I don't think that any of the leads are actually useful in all these four target because the only binding affinity good is for beta styrostol, and that is just at the baseline actually. All the
Yes,
binding affinity should be more than nine, more than there are molecules which actually got more than 12, 14. So you
yes,
can
sir.
see that this binding affinity is very low. Is means uh, I believe that network pharmacology is good to understand the targets and the leads, but you cannot actually rely on this result and utilize the molecules and the target for further study. You should always include a layer of now of AI because it is a predictive and a generative model. It is going to give you information about all this lead and the target. So I will always suggest that try to utilize that as well.
Okay, sir.
Yeah.
Okay, sir.
Okay. Thank you for your
Ramya,
presentation.
uh, hello, one more
Ah,
question,
yes, ma'am.
Ramya. What type of interactions did you notice during your studies? Which type of interactions did you observe?
Hydrogen bonding interaction and hydrophobic interactions also observed, ma'am.
Pi bond
Like,
interactions? Any
yes, ma'am.
pi
Everything is there,
stacking
but
interactions?
mostly I noted, yeah, pi interactions also there, ma'am. But I noted hydrogen bond and hydrophobic interactions, ma'am. Mostly.
Hmm. Oh, okay.
Okay, thank you, Ramya. Now we will move further with our next presenter. So our next presenter is Ankita Sahu. I request Ankita Sahu to start your presentation. And please ensure you present in within time. You have 10 minutes time to present your work.
Hello, hello,
Yes, Ankita.
uh, ma'am. My screen is showing.
Yes, it is visible.
Okay, thank you, ma'am. Uh, Good morning,
Yes.
everyone. I'm Ankita Priyadashini Sahu, a PhD research scholar at Faculty of Pharmacy, Parul University. Today, I will present my topic entitled "With Mitochondrial Dysfunction as a Mutual Mechanism in Multiple Sclerosis and Alzheimer Disease." Multiple sclerosis and Alzheimer disease are clinically different disorder, but they share several underlying pathological mechanisms. Among these, mitochondrial dysfunction has central contribute to the disease progression. So today I'll discuss some common mechanism and all disease, both the disease. First one is the multiple sclerosis. Multiple sclerosis is an autoimmune demyelinating disease of the central nervous system, and where the Alzheimer is a progressive neurodegenerative disorder characterized by the memory loss and the cognitive decline. Uh, both the diseases share common pathological features such as the oxidative stress, neuroinflammation, and calcium dyshomeostasis. Therefore, in additionally, this regulates the mm, calcium homeostasis and the uh, reactive oxygen species. Mitochondria is known as the powerhouse of the cells because they generate the ATP, which is required for the neuronal survival. In addition, mitochondria also regulate the calcium homeostasis and the regulatory oxygen species. Mitochondria dysfunction leads to the neuronal injury and also accelerate the disease progression in both the multiple sclerosis and the Alzheimer's disease. Now, it is the how the mitochondrial dysfunction occurs. First one is the oxidative stress. Due to the excessive reactive oxygen species, it damages the mitochondrial DNA and also the mitochondrial lipids and the protein, which results in the impairing the electron transporting chain and also reduce the ATP production. Second one is the calcium overloaded. Uh, Excess intercellular calcium when enter to the mitochondria through the mitochondrial calcium uniporter. That's result in the mitochondrial swelling and also opening of the mitochondrial pores, mitochondrial pores, and which ultimately activate the uh, apoptosis of the mitochondria. Third one is the neuroinflammation. Uh, Activate the microglia and astrocytes, release some inflammatory cytokines such as TNF alpha, IL-1 beta, and IL-6, and nitric oxide. These inflammatory mediators inhibit the mitochondrial respiration and increase the oxidative stress. Fourth one is the environmental toxins, which includes cupurizin, rotenone, and heavy metals. These these are also responsible for the mitochondrial dysfunction. Uh, Now the pathophysiology. First is the center image is explaining some common pathologies uh, that cause the dysfunction in the mitochondria. Uh, when the neuroinflammation and the oxidative stress is increased and also imbalance in the calcium level, it dysfunction the mitochondria. In the left side, it's showing after the mitochondria dysfunction uh, how the multiple sclerosis occurs. In case of multiple sclerosis, mitochondrial dysfunction cause the ATP depletion that so, due to the ATP depletion, there is an increase in ROS and also imbalance in the calcium. So that due to the imbalance in calcium, uh, oligodendrocytes become apoptosis due to the death of oligodendrocytes. Myelin cannot be maintained due to the loss of myelin sheath. Slowly, nerve impulse and the conductions slow, and axon become exposed and susceptibly to injured due to the axon injury in axon. It also caused the axonal loss that eventually leads to the irreversible neurological disability. In case of the Alzheimer disease, amyloid beta accumulated within the mitochondrial and inhibit the electron electron transport chain complex, which ultimately reduce the ATP production and increase the ROS. Due to the uh, due to the excessive ROS, it also damages the mitochondrial DNA and also the mitochondrial protein. And ATP depletion also impaired the calcium regulation, which lead to the calcium overloaded. Due to the calcium overloaded, it opens the uh, mitochondrial protein transport uh, porter. Uh, that's why it also leads to the amyloid beta and the tau toxicity. Due to this toxicity, um, synaptic failures occurs and cognitive decline. Uh, then the s- since mitochondrial dysfunction is the common to the both the disease, there are some therapeutic targets that used in both the disease. First one is the antioxidant, such as melatonin and the resveratrol, which reduce the reactive oxygen species and also improve the mitochondrial function. And the second one is the mitochondrial targeted antioxidant, such as MitoQ, which is directly accumulate inside the within the mitochondria and neutralize the ROS, which is more effective. And the third one is the coenzyme Q10, which support the electron transport chain that result in the production of the ATP and also protect the neuron from the oxidative injury. Then the fourth one is the uh, elampipyride. Uh, it stabilizes the mitochondrial membrane, uh, which improve the energy production and reduce the apoptosis. The overall these
Ankita,
therapy. Yes, ma'am.
you have only more 3 minutes to present.
Okay, ma'am. This last slide is. Overall, these
Okay.
therapies aim to restore the mitochondrial homeostasis and also reduce the inflammation and slow the neurogeneration in both the multiple sclerosis and Alzheimer. In conclusion, we conclude mitochondrial due to the it promotes the oxidative states and the neuroinflammation and calcium imbalance that also leads to energy failure, ultimately leading to the neuronal damage. Therefore, therapies targeting to the mitochondrial functions, representing the promising therapy for the slowing the disease progression and improving the neurological outcomes. These are my reference, and that's all.
Thank you, Ankita. Now we will go with some questions. I request our evaluators to have some questions.
Yes, ma'am.
Hello?
Yes, ma'am.
I'm audible.
Yes, ma'am.
Okay. So, what are the common molecular mechanisms that are associated with mitochondrial dysfunction in both the diseases mentioned?
There are calcium imbalance, some proactive cytokines, and also reactive oxygen species.
Any more? Any minor one? Any minor mechanisms do they?
Uh, Minor,
Other miscellaneous.
uh, like it's due to the pro-inflammatory cytokines that are majorly cause the disease progression.
Okay. Okay.
And
So, one more. Only by targeting this mitochondrial dysfunction, uh, it will provide a common therapeutic approach for both the diseases. Only
yes,
targeting with the mitochondrial dysfunction.
oh, I cannot understand
So,
the question.
by focusing or by targeting mitochondrial dysfunction, as you mentioned in the presentation,
Yes, ma'am.
shall it provide a common therapeutic approach for both the diseases you mentioned in the
There
presentation?
is actually there is no medicine or therapy is approved in the market, but still in the clinical trials. But I am just sorry, I'm just doing some herbal drugs that may be cure the both the disease.
How the dysfunction of mitochondria may contribute towards the development of Alzheimer's
Hello.
particularly Alzheimer's disease?
If
How it contributes?
if mitochondria is dysfunction, then it ultimately reduce the production of the ATP. ATP is actually the uh, key mechanism that every mechanism depends on the ATP. If ATP is reduced, then ROS also increase and also imbalance in the calcium. If calcium production is less, then it also uh, uh, it also cause in the amyloid plexus, and
Okay.
also
What in case of multiple sclerosis? What in case of multiple sclerosis?
in multiple sclerosis, oligodendrocyte also required calcium. If less calcium, it also is also uh, cause death in the oligodendrocyte. If excessive, then it also cause the inflammation around the oligodendrocytes, which is important for the formation of myelin sheath.
Okay. One more, last one.
Okay.
What are the common mitochondrial pathways that are disrupted in both the diseases? Common pathways of mitochondria.
Uh, Common pathway. Uh,
What common mitochondrial pathways that are disrupted in multiple sclerosis as well as Alzheimer's disease?
uh, is related to the production of
Any
ATP.
pathways?
Any part? No idea, ma'am.
Hmm. Okay. Okay.
Thank you, Ankita. Uh, can you comment on the uh, DNA damage? So you are targeting mitochondrial dysfunction. So what will be the role of DNA damage?
Uh, Mitochondrial also. So I have not. No idea.
You mentioned that you're developing some herbal medicine.
Yes, sir.
Yeah. So have you gone through any literature where
So
DNA
now I
damage?
just recently started
Okay.
searching ROL uh, on this topic. So now I don't go through the DNA topic.
Okay. Thank you. Thank you.
Good presentation.
Yes, Santosa sir, any questions from your side?
No, madam, it's good. It was a good presentation.
Thank you, sir.
Okay, thank you, sir. Uh, Thank you, Ankita. Now we will move further for our next
Thank you.
presenter. So our next presenter will be Srinija. Srinija, am I audible to you, Srinija?
Yes, sir. Am I audible?
Yes, you are audible to me.
Okay.
Yeah.
Uh, Good afternoon, respected judges, faculty members, and other fellow participants. Myself, Srini, from Department of Pharmaceutics, SRM called the Pharmacy SRM IST. I'm privileged to present a research entitled QBD driven formulation, characterization, and evaluation of streptomycin nanosponge hydrogel for topical antibacterial application. Let me begin with the overview of a study. So, graphical abstract. Initially, we prepare the nanosponges. By using the emulsion diffusion method. by input of the ethyl cellulose and polyvinyl alcohol with a design space of central composite design. And after preparing the nanosponges, we'll be characterizing it, and further it will be for the final dosage form for therapeutic efficacy purposes. We're converting into an gel, hydrogel purposes, and for sustained release, which will be further evaluated. Also, antibacterial activity will be studied. So the background of the study includes like skin and soft tissue infections, which is caused by the Staphylococcus aureus and E. coli, which is a major therapeutic challenge due to the formation of a biofilm and poor antibiotic penetration. Although streptomycin has a antibacterial activity, it has poor skin permeation and high hydrophilicity, even rapid elimination at the site of action. So, as a result, in a conventional topical formulations, it is failed to maintain a therapeutic drug level at the infection site. So, there will be limiting in the efficacy of treatment. So to overcome this limitation, ethyl cellulose nanosponge incorporated into carbapol hydrogel, which will provide a sustained release, improve skin retention, and enhance the topical antibacterial activity. As shown in the research cap, the proposed study, which includes like enhanced skin permeation, sustained drug release, so prolonged skin retention will be there, so prolonged therapeutic activity and improve the antibacterial activity. Aim and objective. So the primary aim is to optimize the quality by design based streptomycin loadal ethyl cellulose nanosponge hydrogel for sustained topical antibacterial activity. To achieve this aim, as we before told that the objective is way to streptomycin loadal nanosponges by using should be prepared by using emulsion solvent diffusion method, and it will be optimized by using the design space of a central composite design and categorize characterized by using various things and incorporate optimized nanosponges into a carbapol hydrogel, and it will be evaluated for the further purposes like physicochemical properties, in vitro drug release, ex vivo permeation, skin permeation, and antibacterial activity. So to systematically develop the formulation, we are adopted the QBD strategy, and it is to ensure the robust and reproducible formulations. So quality target product profile, which has a overlay work of an plan of an entire research. And here we can say that by using the central composite design with the 13 experimental runs of phase center value, we have critical quality attributes like particle size, entrapment efficiency, in vitro drug release, and also practical yield. And a selection of critical material attributes, that is, we are evaluating the two independent variables like ethyl cellulose concentration and polyvinyl alcohol. So in between, the risk assessment was done by cause and effect, and after optimizing, preparing nanosponge optimization, characterization, of course, evaluation we carried out in the hydrogel purpose. As I before told that we are using the emulsion solvent diffusion method for the formulation purposes. Here PVA, which is a stabilizers and water in the aqueous phase, where drug is a API, and ethyl cellulose, which is a polymer, DCM is organic solvent. So combining these two aqueous phase and the organic phase, we are using a mechanic stirring, so 1000 RPM for 2 hours. We are after dyeing it, we're getting the nanosponges. What we made into a nanosponge in the laboratory are shown in the figure. And CCD optimization, it was evaluated for the two factors like EC and the PVA, which is considered as X1, X2. And as I told that 13 experimental runs and with optimization of the critical quality attributes. And this slide shows about a design space, which is a 3D response plot, which illustrating the critical quality attributes with the interaction effects of an critical material attributes. And this is a figure 2 showing the corresponding 2D counterpart showing the interaction effects of an independent variables, and overall optimization for the streptomycin sulfate and nanosponge formulation is there. It was shown for the only optimized formulation F5 among the 13 experimental runs. And here, experimental validation and point prediction results were observed in the table for the critical quality responses for what we selected, and has a predicted mean and observed values what we done in the laboratory, and it shows lesser than 2% of an error, which shows a high reliability and predictability of optimization validation. So next, moving to the characterization of the optimized nanosponge F5, which is practical, which is basic thing in the evaluation parameters. So here, 84.8% practical yield we got, and efficient reproducible nanosponge preparation process we can up to obtain from this practical yield. And entrapment efficiency for the optimized nanosponge will be 92.6%, which is better encapsulation efficiency within the ethyl cellulose. And as we know that emerging trend is nanotechnology, so the research is also basis on that. So the particle size is the most important characteristics as we observe. So the we assured here is 505 nanometer. So here it will be nano distribution of a particle size and zeta potential of negative 33.6 milliwatts, which has high stability, high colloidal stability for in the electrostatic repulsion purposes. And moving to the thermal analysis, it is done with differential scanning calorimetry. Here we observed for the pure drug and for the formulation nanosponge. Pure drug, there will be an crystalline peak, and whereas in the nanosponge, we observe that there will be reduction and disappearance of a crystalline peak, what we observe in the drug, so that there will be a transition state to the amorphous form. It ensures that the drug polymer compatibility. And moving to the morphological analysis, since it is a nanotechnology, we came here to for the porous structure, and spherical structure is shown in the figure 6. And porous structure architecture supports the good drug loading capacity and the sustained drug release, and in vitro drug release followed by the characterization. So here I have shown the graph for all 13 experimental runs, but for the formulation optimized 5, which is the highest highest drug release were carried out for the 8 hours of time, and highest percentage was cumulative drug in vitro drug release were observed, so that we ensure the sustained release of the sustained release profile. And it says like it also ensures that diffusion controlled release of streptomycin, which is encapsulated in the polymer like EC. And after the optimization, next made into a final dosage form by incorporating that into one nanosponge hydrogel. Specifically, carbopol 934 grade hydrogel was used because of inconsistency to make as an hydrogel. And their additional ingredients were used in this nanogel were propylene glycol, which is penetration enhancer, triethanolamine neutralizer to maintain the pH, and distilled water as a vehicle. And physicochemical evaluation, which includes pH of about 6.21, which is suitable for the topical application. Drug content, which has uniform drug distribution, and it is achieved 84.27%. Viscosities of about 7357 centipores, which is about for the consistency for the topical use, and spreadability was good, and organoleptic properties and characteristics for order was good. And next, evaluation for the nanosponge hydrogel specifically is done for the 8 hours too. So here the in vitro drug release study for nanosponge hydrogels for 8 hours was shown in the graph, and it was fitted into the various release kinetics. With zero order, first order, Higuchi and Coase-Meyer peppers as a release kinetic profile. Its best fitted model was Coase-Meyer peppers, and with the absorption rate of 0.971 with a half square value, and follows a non-frequent diffusion, which is suitable for the nanosponge hydrogel, and ensures a sustained release too. And further, for the main aim was to the antibacterial activity, and it was done for 2 different organisms like E. coli and Staphylococcus aureus, where E. coli is a gram-negative bacteria, whereas Staphylococcus is a or Staphylococcus aureus is a gram-positive bacteria. We all know that streptomycin is basically effective against the gram-negative bacteria, so it is a well shown a zone of inhibition rate in the against E. coli compared to Staphylococcus aureus, and it also shown with Staphylococcus aureus, but not such efficient one. And with a significance of lesser than 0.05, it was done by the method called zone of inhibition. And moving
Hello.
to the yes, ma'am.
Yeah, Srini, you have more 2 minutes to present, so present.
Okay, ma'am, I am able to complete it. And moving to the X fiber permission study, your X fiber permission study is done by the excised skin of a goat to be compared to the plain gel with nanosponge hydrogel. Nanosponge hydrogel, which has about 80.2 microgram per centimeter square. And for stability studies, we are done for two different temperatures, 25 plus or minus 2 degrees Celsius and 40 plus or minus 2 degrees Celsius, and with a period of 90 days, and it does not make any major significant changes. In conclusion, quality by design based streptomycin loaded ethyl cellulose nanosponge hydrogel was successfully developed and optimized by the nano particle size zeta potential entrapment efficiency. They demonstrate excellent physicochemical stability. And for the in vitro drug release, we observed the sustained drug release for 8 hours of time. And significant antibacterial activity was against E. coli and Staphylococcus aureus. Overall, it shows a sustained topical drug release system for the effective management of bacterial skin retention. And future scope, we are planning to conduct in vivo preclinical studies for the safety efficacy and long-term stability studies under the ICH guidelines under the clinical evaluation for to establish safety effectiveness in the patients. And additionally, it would be scale up and commercial development if it's all successful. And my research work was connected with the SDG goals like three, good health and well-being, and industry innovation and infrastructure, and 12 is responsible consumption and production. So I would say like overall. A study demonstrates that how nanotechnology and systematic pharmaceutical development through QBD can contribute to the next generation topical antibacterial therapies, so that aligning with the current emerging trends in healthcare science and technology. So, with this, I conclude my presentation. Thank you for your kind
Thank
attention.
you, Srini. Now we will go
Yes,
with some questions. I request our evaluators, Santosha sir and Anita ma'am and Praveen Bade sir to have some questions.
Excuse me. Hello.
yes, ma'am.
Am I audible?
Am I audible?
Audible.
Yes, ma'am. Yes, ma'am.
Okay. How does the QBD approach improve the formulation and optimization of the nanosponges of streptomycin for the antibacterial action by applying topically? How this QBD approach improves the formulation and optimization?
Yes, ma'am. QBD is basically we build the quality into the formulation itself so that it can make the quality and efficacy of a product so easily, and by reducing the chemical wastages, and also by concluding with various evaluation parameters. And especially for the streptomycin, it is an antibiotic category, aminoglycoside 1, which is already proven in the articles like it will be provide an antibacterial activity. Further, the problem is to penetration only, so that we can encapsulate, and with other nanotechnology technique, we are proving it, ma'am.
How this formulation, hydrogel, nanosponge hydrogel, in enhancing antibacterial activity and also control the release of the drug from the topical application? How effective this one to enhance the antibacterial activity?
Ma'am, it is for the over 8 years of study, so that antibacterial activity shown by sustained release only. So it is not overly the controlled release. So sustained release is achieved by the encapsulation of streptomycin in the ethyl cellulose, so that it will be in the hydrogel formulation. After applying it to the skin, it will be releasing the ethyl cellulose polymer after penetrating into the stratum corneum layer. So it will enhance antibacterial activity when the wound is in a topical application.
So one more, what type of parameters, evaluation parameters used in the assessment of quality as well as the stability and also efficacy of the streptomycin nanosponge hydrogel? What
Ma'am,
are the key
till
evaluation
now,
parameters?
evaluation parameters, what we done till now is for the homogeneity, for hydrogel, I'm saying, homogeneity, pH, and observing some of the changes, like basic thing what we done, we can do in the laboratories we run. And furthermore, particle size we're planning to observe, ma'am.
How you evaluated antibacterial activity? Any
By
method
zone of
you perform?
by zone of inhibition method, ma'am.
zone of innovation method. No?
Yes,
Only
ma'am.
one method you performed. One model.
Sorry, ma'am.
Only one model you performed in the
Yes,
lab.
ma'am. Yes, yes, ma'am. Yes,
Evaluation.
ma'am. Yes. Only
Okay.
one model, ma'am.
How many gram positive and how many gram negative species you used in the method evaluation? How gram positive? How many gram positive and how many gram negative strains you have taken for the evaluation?
Ma'am, I forgot about this. Sorry, I don't about the strain's name, but only one strain was used for both thing.
Only
And
one strain.
following, ma'am,
Only one strain you used, huh?
one strain is used for E. coli. For the other strain, we planning to perform. Other strain was
Okay.
used, but we don't find the effective one. So Staphylococcus aureus, we don't find the effective strain. So we're planning to do it again. So yeah.
Okay. Okay. Thank you.
Thank you.
Thank you. Hello, can you hear me?
Yes, sir. Yes, sir.
Yeah. Thank you for your presentation, Shirini. There is one
Thank you.
question. Why you are preparing the sponge, nano sponge? Why not any other nano formulation?
Sir, nanosponge has better encapsulation efficiency when comparing to other formulations. It has and wide range of drug loading capacity too, and we can easily incorporate it into when topical administrations like gel, so that easy applications for the patient to use. We planning for it. So.
Okay, so do you need to do clinical studies for nano sponge?
Yes, sir, but preclinical studies were fully clarified, we can go with clinical studies because streptomycin sometimes have the toxicity range like irritations studies, all those things. So first, after clearing the preclinical studies, we can move with the clinical one, and it is necessary to do for nanosponge also for the gel.
Okay, and are there any formulation in the market with nano sponge?
Still now, I don't find any proper formulation, sir. Conventional topical only I have seen referred one, but it is a emerging trend, so that I was finding to solve it.
Okay, okay. Thank you. Thank you.
Thank you, sir.
So, yes,
Hello.
Santosha sir, do you want to?
Yeah, good presentation. All the best.
Thank you, sir.
Okay. Thank you, sir, and thank you, Srini. So now we
Thank you,
will
ma'am.
further move to our next presenter. So our next presenter is a Trivedi Aishwarya. I request you, Trivedi Aishwarya, to present your work, but ensure you have 10 minutes to present your work.
Okay, ma'am. Am I audible to you? Hello?
Yeah?
Hello? Am I audible to you, ma'am?
Oh, yes, you are audible. Yes, you are
Okay.
audible.
So, good afternoon, everyone. So, I am Trivedi Aishwarya, Assistant Professor at the Kamdar College of Pharmacy, Rajkot. So, my topic for the presentation is formulation and evaluation of the lactoferrin nanoparticles loaded in situ gel for the post-menopausal osteoporosis. So, these are the contents: introduction, rational aim, objectives, review literature, materials, methodology, evaluation, and conclusions. So, introduction. What are the post-menopausal osteoporosis? So, according to WHO, 1 in 2 post-menopausal women have osteoporosis. So, osteoporosis is the skeletal disease which is characterized by low bone mass and microarchitectural deterioration of the bone tissue, with the consequences of the increase in the bone fragility and susceptibility to the fracture after the menopause. There are two types of menopause: menopause related or the age related bone loss. So, the menopause related bone loss gives the 3 to 5 years with the rapid bone loss but in the age-related 10 to 20 years with the slower bone loss. Mainly trabecular bone affected in the menopause, but in the age-related involve cortical and trabecular bone. So it is characterized by bone mineral density. It is the 2.5 standard deviation more below the mean of the young adult range. So major postmenopausal problems are the bone loss, weakened immune system, bacterial and vaginal infection, vaginal dryness. So these are the conventional treatment available for the postmenopausal osteoporosis. These are the oral tablets, capsule, or oral jelly available. But it has some problems like gastrointestinal tolerabilities, esophagitis, esophageal reflux. Need to be taken 2 hours after or before the meal. Need to be taken with a large amount of water. And for the intravenous, it gives the flu-like symptoms, pain in the joint, muscles, fever, and headache. So we are preparing the lactoferrin nanoparticles loaded in situ gel for vaginal delivery of the biphosphonate. So lactoferrin is the multifunctional glycoprotein for the transferrin family, which has the activity like antimicrobial, antiviral, immunomodulatory, bone regeneration. So we are preparing the nanoparticle with the lactoferrin as a polymer. Uh, and the biphosphonate. These are the drugs which we will use for the bone regeneration property, which gives the slow down the bone resorption by the reducing osteoclast function, improve the bone density, and reduce the risk of fracture. So biphosphonate given the vaginal route reduce the side effect related to oral or IV routes. Also directly goes into the systemic circulation. We can deliver 10 to 30 times more biphosphonate by the vaginal route. Then we will go for the in situ gelling approach. These are the importance of the in situ gelling approach, like the mucoadhesion at the vaginal site. So it gives the vaginal dryness reduction and the increase the retention time of the drug. These are the rationale for the formulations. The oral biphosphonates have this, some side effects. So we can go for the vaginal route that directly goes into the systemic circulation, so avoid the first pass metabolism, gives sustained release effect. Also lactoferrin is a polymer, also has the bone regeneration, antimicrobial, antiviral, and immunomodulatory effect. Then reduce the postmenopausal dryness. These are the major factors which affect the postmenopausal woman. Then the patient compliance and easy for the applications. These are the aim and objectives. Formulation and evaluation of the lactoferrin nanoparticles loaded in situ gel for the vaginal delivery of the biphosphonate for the postmenopausal woman. For the objectives, we are first preparing the lactoferrin nanoparticles. Then we will go for the pre-formulation study, drug excipient stability study, then optimize the formulation by QBD approach, then characterize an evaluation of these lactoferrin nanoparticles, then optimize the batch formulation with in situ gelling approach, then characterize evaluation study in situ loaded lactoferrin nanoparticles, and to perform accelerated study by preparing formulations. These are the plan of work. We first perform the pre-formulation study by the calibration curve, DSC, FTIR, then drug excipient study, then preliminary, then screening of the factor by the plaquette Berman design, then optimization by the nanoparticles by the box bank and design, then evaluation of the optimized batch incorporated into in situ gelling system, then evaluation of the in situ gel, then container related evaluation and short-term stability study. These are the material: drug alendronate, polymers, lactoferrin for the nanoparticles, carbopol poloxamer for in situ gelling, then surfactant, cross-linking, solvent, and others. These are the drug profile. These are the methodology we are used for the pre-formulation study. First is calibration curve, then identification of drug by the DSC FTIR, excipient stability study. Then we go for the QBD approach. First we select the QTPP elements, then selection of CQA elements, then Ishikawa diagram. So these are the QTPP elements which affect our formulation. So physical attributes, color, odor, appearance. In the dose, we are selecting the dosage form, morphology, category, route of administration, and the performance by the packaging or stability. For the CQA of the nanoparticles, we are selecting 3 factors: particle size 150 to 400. 150, below the 150, it directly goes into cervicovaginal mucus and not give as much as effect. above the 400 nanoparticle, it is difficult to diffuse in the vaginal mucosa, so we will target 150 to 400 nanometer. For the entrapment efficiency greater than 70% for good effect, and drug release 70 to 100. These are the Ishikawa diagram. The factors which directly affect our formulation throughout this study, these are enlisted over here: formulation related, method related, environmental related, process related. Then we will go for the nanoparticle formulation. From these three, we will go for the dissolution methods. For the preliminary screening, we are taken these factors and the values. From this, we will go for the Pluckett-Berman design. These are the factors: lower concentration and higher concentration. For the dependent variable, we will take the particle size, PDI, and yield. These are the layout of the Pluckett-Berman design. Then we will take the major factor which affect from the Pluckett-Berman design for the Box-Benken design. These are the concentration of polymer, flow rates, and the speed, which highly affect our nanoformulation. So we will take for the Box-Benken design. Then the dependent variables are the particle size, entrapment efficiency, and drug release. These are the layout of the Box-Benken design. We will prepare the batch according to this. Then we go for the evaluation of the nanoparticle like yield, particle size, zeta potential, polydispersity index, entrapment efficiency, in vitro drug release study. Then we will go for the in situ gelling approach. For this, we will take the chitosan, carbopol, and carbopoluronic concentrations. Then we will go for the nanoparticle loaded in situ gel. We will evaluate viscosity, gelation temperature, gelation time, pH, and in vitro drug release study by the France diffusion cell. And lastly, we will go for the container related evaluations and the stability study. These are the result. Calibration curves gives the R square nearby 0.99. These are the FTIR of the drug and the standards. So we will give the functional group peaks according to the standard. These are the DAC of the alendronate sodium. So we will get the melting point as such as reference. These are the FTR physical mixture. So we will get the physical mixture and we will get the peak. So it gives the information that there is no interaction between the alendronate sodium and other ingredients. As such as FTIR, DSC also has no interactions. In the preliminary study, we will select the dissolution method because the particle size is lower than the other method. Then we will select the organic solvent according to the particle size, then surfactant, polymer concentrations, stirring time, stirring speed, and surfactant concentrations. Then we will go for the Pluckett-Berman design. These are the data which gives the particle size, PDI, and yield. From this, we will select these three by the Pareto chart: concentration of the polymer, flow rate, and the speed, which highly affect the formulation. So we'll go for the Box-Benken design. From this, we will get the particle size, entrapment, and drug release study. Then we will go for the in vitro drug release study, which gives this in 6 hours. It gives the highly. Then we will go for the checkpoint batch method. In this, the predicted value and experiment value are relatively nearby same, so we will go for the optimization batch. Then in vitro drug release study, the optimized batch nanoparticles, it gives the 83.82% after the 6 hours. Then we will go for the in situ gelling system. We will select the carbopol and chloronic for the in situ gelling system because it has the higher viscosity. These are the evaluation parameter of the in situ gelling system: viscosity, chelation time, chelation temperature, and pH. These are the container related evaluation. We will perform in this spray pattern. So we will give the actuation number, volume upon each actuation, and drug content for the actuations. These are the short-term stability study. It gives that after this stability study or before the stability study, there is no major changes in the formulation. So it is the stable formulations. These are the conclusion. The vaginal delivery of the biphosphonate in the form of the nanoparticle contained in situ gel has the advantage over the oral and IV because it eliminates the side effect. Also, alendronate has the bioavailability 0.5 to 2%. So, by applying the nanoparticulate or vaginal route approach, we can improve the bioavailability or gives the sustained release effect. These are the references. Thank you.
Thank you. I request our evaluator to have some questions.
Okay. Hello, am I audible?
Yes, ma'am.
How it helps in managing the post-menopausal osteoporosis?
What, ma'am?
How it helps in managing post-menopausal osteoporosis? Lactoferrin, your
Generally,
formulation you mentioned now. How it manages menopause and osteoporosis?
generally, in traditionally there is no any other formulation or anything available. But this is according to WHO. This is the serious condition. So we have to approach that. Okay,
त्याची
we will
PPT टाकली
find
नाही अजून.
some formulation which is directly give the effect of postmenopausal woman. Generally, we will take the vitamin D or calcium or anything else like this. But no proper treatment are available.
What are the
Or
pre-formulation
available.
parameters you studied? Pre-formulation parameters.
Generally, we are performing particle size PDI in vitro drug release study ex vivo by France diffusion cells.
And what are stability parameters or conditions in your study? Stability studies.
For this, we have kept it for
Stability
30
parameters or conditions.
particle size and trapid efficiency on drug release because we are formulating nanoparticles. So majority are the
No
particle size
temperature?
and trapid. Yes, ma'am.
No temperature is mentioned. You mentioned in one slide, na? Temperature you mentioned in one slide, I think so. How
In
is
the
the temperature
description, I have
affecting
told.
the stability?
In description, I was told that the formulation was kept at 30 plus or minus 2 degrees centigrade for the 1 month.
Per period?
1
month.
month.
And what are the surfactants you used in your study? Surfactants.
380.
390. Any other, can you name other excipients in your study? Just the names of excipients.
Triethanolamine for the pH adjustment we are using because we are going for the vaginal route. So we have to check whether the pH are appropriate to vaginal formulations or not.
Okay. You mentioned short-term stability study. You mentioned, na? What is the short-term? What is the short-term? That means what is the duration of that
For the
study? Stability study.
generally they are performed for the 3 months, but due to the lack of timing for the project, we are performing for the 1 month only.
One more, last one. FTIR you mentioned, na? FTIR. Which functional group you identified? Which functional
What,
groups
ma'am?
in FTIR? FTIR.
NH group. These are the majority
NH.
in the our.
And only that one you identified?
No, ma'am. ROH group, PWO bond.
Double one. ROH is there. Okay. In mixture also, no?
Yes, ma'am.
In mixture, you mentioned in the mixture. FTIR of the mixture you mentioned in next report, next slide. In
Yes,
that
ma'am.
also you identified OH, PO. In this you did not mention the group.
No, ma'am. I am not
Same group in the
mentioned.
mixture also. Physical mixture, yeah. In the physical mixture, FTIR of physical mixture you mentioned in that slide.
Yes,
In this
yes, ma'am.
also,
But functional group,
in you
I have not mentioned the functional group.
but according to the standard, it will give the same peak at the our drug has.
Okay. Okay.
Thank you, ma'am.
Thank you for the presentation, Trivedi,
Yes,
Aishwarya.
sir. Yes, sir.
Uh, just a couple of questions. So you have prepared nanoparticles, right?
Yes, sir.
So what is the purpose? Why did you prepare nanoparticle and not went for a normal formulation?
Because, sir, we have selected the drug alendronate. It has the 0.5 to 2% only bioavailability.
Okay.
And the other formulation available like oral and IV has the major side effects. So we are preparing the formulation of nanoparticles which eliminate the side effect also, and also it gives the better bioavailability than the formulation available in this market.
Okay, and what about the regulatory and clinical trial? Do you have to do
No,
clinical
sir, these
trials
are the
and pass the
future.
regulatory to bring the product in the market?
Sir, these are the feature scopes we will perform after giving the permission by animal ethics committee. We will perform the animal stability study, animal study by the giving the permission after.
Okay, are there any nanoparticle formulation in the market?
Mm, no, sir. Generally, IV or oral tablets available.
No, any nanoparticles. I'm saying not the disease which you are targeting. Any.
Yeah, yes, sir. For the cosmetics, also there were some nanoparticles. Shampoo also has the same nanoparticles formulation. Like L'Oreal has
No,
shampoo.
no. See, the regulatory is different for cosmetic. Regulatory is different for the formulations, allopathic. It is different. I'm asking
Okay.
for your project. If I'm using a molecule which is already a allopathic drug and preparing any nano
Mhm.
formulation, can it go into the
Okay.
market? Have you actually studied or identified any of the product in the market which is
It might be difficult.
developed? Yeah, so how means my thought is this: after doing all this, if it is not going to be utilized by the patient, how does this project will create a value and contribute in the healthcare?
Yes, sir. It will be difficult, but after going for the animal study, if it gives the better result, then it might be
Yeah, but I think
go for
you
the
need to
production.
understand the regulatory and clinical perspective of the formulation as well, isn't it? Means animal
Yes, sir.
studies is part of research. It is a preclinical work. You will get, permission and it will be done. But what about if the product is not going to pass the clinical studies because of the regulatory? How it will be utilized? That is my concern.
Okay, sir.
Yeah. Okay. Thank you. Thank you for your presentation. Nice work.
Thank you, sir.
Thank you, Trivedi Aishwarya. So now we will move further with our last presenter of our session. So the last presenter of our session is Manasi Pavaskar. Uh, I request Manasi Pavaskar to present your work. Am I audible to you?
Yes, ma'am. Is my sound clear?
Yes, you are audible to me. You
Ma'am,
will have 10 minutes to present your
yeah,
work.
ma'am, do I have to keep my camera on or is it okay if
It's
I
up
keep
to
it?
you.
Okay.
Yeah, it's up to you.
Just because of the bandwidth, I'm keeping my camera off. So, good evening, everyone. My name is Dr. Mansi Pavaskar. Today, I'll be presenting a study which was done by my MSc student. It's entitled Phytochemical and Pharmacological Evaluation of Rhizomatous Plant Species from Goa. I hope the slides are visible. I'll quickly move to the introduction. So rhizome, as we all know, it's an underground stem, okay, which spreads horizontally and it has major function in storing the nutrients and aiding the plant for its propagation. So apart from this job, it has a variety of biochemicals, okay, which actually help for the growth of the plant. However, these biochemicals also have advantage to the humanity. Now, India is so known for its diverse flora and fauna, including the various medicinal plants that we use for against various diseases or any disorders. So our study tried to focus on application of these rhizomes as a for its medicinal properties. So we have focused on the ribosomes which are found in Goa, okay, the Keri region of Goa, and then we have worked upon it. So this is a brief literature review which talks about the rhizomes which have been used all over the world or which have been studied all over the world for its various properties like anti-inflammatory, antioxidant, anti-cancer. So keeping this in mind that rhizomes do have potential, we went ahead with the collection of the rhizomes. We selected five different rhizomes: crab ginger, sweet flag, mango ginger, butterfly ginger lily, and Indian head ginger. So extracts, methanolic extracts were prepared from this rhizome and then pharmacological and phytochemical analysis was done. Major focus was kept on pharmacological analysis and phytochemical was just an additional thing. So coming directly to result and discussion. So as I said, these rhizomes were collected from Goa, from the Keri region of Goa. And this picture shows the plant and the rhizome which we have selected from the study. The first being the crab ginger and then Indian head ginger is the last one. So in the top row you can see the plant and the bottom row shows the rhizome of the plant. After collection of the rhizomes, these rhizomes were nicely washed, dried thoroughly, and then powder was made of these rhizomes. And this from this powder we prepared the methanolic extract. Okay, we kept the concentration as 100 mg per ml so that the concentration remains uniform throughout the tests. The image shows over here the extract which we obtained from the various rhizome powders, the methanolic extract of various rhizome powders. Then we first test that we did was antibacterial activity by well diffusion method. All the five rhizomes were tested against three pathogenic genus, which is Escherichia, Shigella, and Vibrio. And among the five different rhizomes, we saw that crab ginger and Indian head ginger showed activity against all the three pathogens. However, we couldn't get any result in case of sweet flag and butterfly ginger lily. Mango ginger also showed activity but only against Escherichia species. The control was the methanol which was used to prepare the extract. Next was the anti-inflammatory activity. For this, we used the HRBC membrane stabilization method. Basically, we were checking the ability of this extract to prevent the inflammation of the human RBCs by stabilizing the membrane. So, the standard solution which was used over here was aspirin. So, as compared to the aspirin, sweet flag, mango ginger, butterfly ginger, and Indian head ginger showed better activity. Crab ginger also showed the activity, but it was less as compared to the standard. And mango ginger showed the highest percent of protection against the inflammation. Next was the antioxidant activity by DPPH radical scavenging method. Here again ascorbic acid was used as a standard solution and Indian head ginger showed the highest DPPH scavenging effect. That means it showed the highest antioxidant activity. Next, we tested this extract for anti-arthritic activity. So this was an indirect test. So we see that in case of arthritis, we see a lot of protein denaturation. So any sample which is able to inhibit this protein denaturation will be able to act as an anti-arthritic agent. Okay, so here we had used a synthetic drug, diclofenac, as a standard solution. All the rhizomes showed the activity, however, the result was not comparable to the standard solution. the inhibitory activity of the standard solution was way higher than all the five extracts. Next was the anticoagulant activity by the prothrombin time test. So basically, here we check the clotting of the blood, and here the positive control was the EDTA, which so basically the time taken for coagulation was recorded in minutes, and the positive control, which is EDTA, took the maximum time, showed the maximum time. However, comparative results were also shown by all the five X-ray of the rhizomes. Okay, next that was the pharmacological assays. Then we went to the phytochemical assays just to check the basic composition of the extracts. So we tested for tannins, flavonoids, saponins, steroids, and phenol across all the five different extracts. Now I'll tell the results in the summary. So as I said, five different rhizomes were selected for this study, being crepe ginger, sweet flag, mango ginger, butterfly ginger, lily, and Indian head ginger. And we wanted to check their medicinal properties. So Indian head ginger and crepe ginger showed the bacterial activity against fly, Shigella species. Just excuse me, there is some issue. Okay, sorry. Indian hair ginger, crab ginger showed the antibacterial activity against E. coli, Shigella, and Vibrio, whereas mango ginger exhibited only against Escherichia species. Then all this extract showed the anti-inflammatory potential, with mango ginger exhibiting the highest percentage of protection, which was much higher than the standard aspirin. For antioxidant test, all the extracts showed the positive results, out of which Indian hair ginger exhibited the highest scavenging effect, followed by butterfly ginger. And both of these showed the higher activity than the standard ascorbic acid. Then the inhibition of protein denaturation for anti-arthritic potential was studied, and inhibition was shown by all the extracts. However, it was very less as compared to the standard diclofenac sodium drug. Then finally, all the extracts exhibited notable anticoagulant efficiency, with the sweet flag extract resulting in the prolonged coagulation time of human plasma as compared to the other extracts. The phytochemical composition revealed the presence of flavonoid but absence of phenols. Tannins, steroid, and saponins were present in crepe ginger, Indian head ginger. Additionally, steroid was also present in butterfly ginger lily. So coming to the conclusion, after studying all these five rhizomes, we came to a conclusion that these rhizomes have medicinal properties and they have potent activities which needs to be further studied or optimized. Okay, so the findings established the antibacterial activity, anti-inflammatory activity, and antioxidant potential of these rhizomes. Also, we were able to see the inhibitory activity against protein denaturation and anticoagulation properties. So, with further studies and detailed studies, will be able to formulate a drug which can be used in healthcare or pharmaceutical. So, these are the references which are used in the presentation. Thank you for the time provided.
Thank you, Manasi. Now we will have some questions. I request our evaluator to have some questions.
Hello.
Yes, ma'am.
Okay. So you presented different types of phytochemical constituents, now.
Yes.
How to contribute towards the pharmacological activity of the species? How those constituents may contribute for the pharmacological properties?
So, ma'am, so what we are suggesting is that currently this is a very preliminary work, but if we are able to identify specific compounds which are showing these anti-arthritic, anticoagulant activities, then we can use it in the healthcare for against various diseases, or even, for example, clotting disorders or any heart-related disorders. But for that, we have to identify specific compounds which is contributing to it. So this was a very preliminary study which was done only using the extracts. Detailed study will help us to target the various disorders in a better manner.
Okay. What solvent used for extraction? Only what solvent used?
So, ma'am, the literature showed that methanol is able to extract most of the potent
You
biochemical
used ethanol only.
methanol.
Any combination? Any combinations used? Only ethanol used.
No, only methanol was used, ma'am. No combination was tried. We just
As
depended
per
on
literature,
the,
you followed.
yes,
As per literature, you followed.
yes.
You can change. You may try as a trial.
Yes, ma'am. We can try different solvents as well as ratios could be changed to get a better extraction of the biochemical molecules.
Okay, one more. Which pharmacological studies you performed in your lab? Any in vitro models are there?
No, ma'am, no models were used. So, as I said, this was a dissertation work. So, whatever was feasible in that time, we could only establish the potential of the extracts. However, they were not tested.
Special. General గా చెప్తారు. And which is used as standard for anti-inflammatory activity?
Sorry,
You
ma'am. For
already performed anti-inflammatory activity, na? What is
aspirin
the
was
standard
used.
used in the method? Anti-inflammatory method?
Aspirin, aspirin was used, ma'am, as a standard.
For
Once
antioxidant activity?
ascorbic acid.
Ascorbic acid.
Yes,
Okay.
ma'am.
And last one, can you tell the compound which is responsible for majority of the activities you majorly contributed towards the activity?
No, ma'am, we couldn't pinpoint to any compound. As I said, we didn't do any detailed study. So only we couldn't pinpoint to any specific compound in this study.
Okay. Thank you.
Thank you, ma'am.
Thank you, madam, for the presentation. So this was a PG biotechnology student, right? Work
No, sir.
was done.
This was a PG environmental science student.
Environmental science.
Yes.
Okay. And are there any in vitro model in your university which you are
No,
planning
sir.
to do?
Currently, no, sir. Matlab, I have not done any further work on this aspect till now. This was done a year ago, almost last year. This was done. So this year we are hoping to take ahead the research, and then, as ma'am suggested, if we can pinpoint a compound or extract a compound, that will be really help us to establish this work.
Okay. Because it looked like just the beginning of a project
Yes, sir. It was
where
actually.
where actually um, there is no relation established against activity, and also the relation for the molecules or maybe developing a formulation is not established. So I think the work is good. You already got a base there, but you need to decide, do you want to actually increase the uh, quality studies, quality control studies, or do you want to go for formulation or activity? Because all these, because with the plants, there are already so many activities are already been reported. Definitely, in your study, the uniqueness is that you have taken the species from a area which is there in Goa, which can again, establish the longer relationship and actually have some unique molecules in those plant. But at the moment, it is just the beginning. So yes, work is good. Thanks. Thank you.
Thank you, sir.
Uh, Thank you, Manasi. Uh, Thank you to all our presenters and evaluators too.
Okay, ma'am. The session is completed, ma'am.
Yes, ma'am, the session is complete.
Shall I leave?
Yes, ma'am.
Okay, thank you. Thank you, ma'am, for giving the opportunity. Okay.
Thank you, ma'am. Thank you to all presenters, and we sincerely thank all evaluators for their valuable contribution to the conference. With this, we have successfully concluded all the scientific sessions scheduled for day 1 of Swalife Biotech annual 4th Annual International Conference 2026. Today's conference has provided us with remarkable insights into predictive intelligence, sustainable development, and innovative poster presentation and outstanding oral reach presentation. As we conclude the first day of Swalife Biotech 4th Annual International Conference, it is my privilege to propose the vote of thanks. On behalf of the entire organized committee, I extend our sincere gratitude to our keynote speaker, Dr. Praveen Bade sir and Dr. Nilesh Narayan Prasad sir, for sharing their valuable knowledge, experiences, and visionary insights. Uh, your inspiring sessions has our perspectives and motivated us to excellence in scientific research and healthcare innovation. I would also like to express my heartfelt appreciation to all our poster and oral presenters. Your dedication, hard work, and commitment to advancing scientific knowledge have made today's conference truly enriching. A special note of thanks goes to our respected judges to their time, thoughtful evaluation, and encouragement to our participants. We are equally grateful to our organizing committee, technical team, volunteers, and everyone working behind the scenes whose efforts ensure the smooth execution of today's program. Finally, we sincerely thank every faculty member, researcher, student, and participant for your presence and active engagement throughout the day. We look forward to welcoming to all tomorrow for another exciting day featuring expert lectures, oral presentation, the awards ceremony, and the Savitribai Phule Best Faculty Research Award. Thank you, everyone.
Thank you.